All right, what's up, everybody? Welcome to Jupiter Brain. Today, we are super stoked to have the one and only Dr. Waters on a podcast that we've been looking forward to for
pretty long time.
Yeah. So I'm super stoked. I knew from the moment I met you. So I was sort of introduced. Not necessarily to you, but about you by Dr. Lyons. And so I was talking to Dr. Lyons about this sort of quest that we've been on for about 3 semesters now, which is to try to get a good list of all the research going on at UCO. And she told me, well, you should go talk to Dr. Waters, because she's sort of, and so maybe you can clarify here, are you the head of this Tri-Center Symposium, or whatever it's called?
Yeah, so not the head, but in the College of Math and Science, we have 3 different research focuses. One is called CIBER, and it's the oldest and the biggest of the research centers, and that's the one that I'm the director of right now, and so that is the Center for Interdisciplinary Biomedical Research and Education.
Mouthful.
A mouthful. That's why CIBER sounds so much better.
Yeah.
And then the one that Dr. Limley and Dr. Lyons head up is going to be CREIC, which is more of the supercomputing, and don't put me on the spot to remember the acronym, but it's supercomputing, has to do with Buddy.
Yeah.
And then the last one has to do with forensic science and wildlife, so that's conservation and CFACS. And Dr. Jackson heads that one up. So I'm part of a larger team that does this, but CIBER being the biggest of the 3, we tend to have a handle on a lot of the biomed education and research going on.
Gotcha, cool. So, yeah, it's funny because, ever since you said yes to this, and like I said, I knew after that first conversation we had, and I asked you literally after we got done talking, I was like, would you want to come on the podcast?
And I was so excited to, and my schedule has been so crazy, so and then the snow day, but I'm so glad to be able to do this. I'm so excited to talk to you guys.
Yeah, we going through college now and trying to set up meetings with professors and stuff like that, especially ones who, have big roles like you do in the college. We understand that you guys get a little busy sometimes.
We do.
We're just so privileged to have you and other professors come on. You're our 3rd professor for us to get to interview now. What we'd like to plan on talking about today is a little bit of your background, sort of how you got into research, maybe a little bit about what your research is about. I really would love to know a little bit about why you made the transition from the NIH to UCO, and maybe a little bit about the courses you teach. So just do a little background for you, so you don't have to explain everything, right? So, Dr. Waters, you have your bachelor's in chemistry, right?
Yes.
And your doctorate in
Pharmaceutical Sciences.
Pharmaceutical Sciences.
Yes. With an emphasis in pharmacognosy, which I'll talk about later.
Yeah, and so, so I saw that you're from Altus, which is, I'm a fellow born and raised Oklahoman, so really cool to see that. And I was looking at Altus today, and it's a pretty good drive from here.
2 hours and 20 minutes if you're doing it right. So, I grew up as a military kid. So Dad was 24 years in the military. We lived everywhere. The last base he decided was Altus, and that's where he retired. And that's where both my sister and I graduated high school. So not necessarily full native Oklahoman, but I love it, and I claim to be from here because I've been here the longest. So.
Awesome. And so some of the classes you've taught, which I would love to get into definitely one of them. So you've taught General Chemistry 1, General Chemistry 1 Recitation and Laboratory. So that's just the lab of chemistry one, right?
Yeah.
And then General Chemistry 2, Theory and Practice of Nuclear Magnetic Resonance. That's the one I would like to kind of pick your brain about.
Yeah.
And then also Seminar in Medicinal Chemistry, which, speaking of medicinal chemistry, we're actually right across the way from the chemistry section in the library.
I actually had come out of the elevator on that side, and I happened to look left, and I went, that's a mass spectrometry book, and so I had to stop and browse for a second. So I would have been here a few minutes earlier, but it math aspect caught my attention.
That's totally fine. We're both super nerds, so we get it. We get distracted by the same stuff.
Yeah. So I guess kind of start with the journey, if that sounds good. Or you want to classes, or?
What we always like to start with here at Jupiter Brain is we like to ask sort of how your week's been.
Oh, yeah.
But actually, if you could maybe talk about how the beginning of the semester's been for you.
Yeah. It's always a crazy busy time. I think especially when you're in academia, you make that jump because you love the students and you love interacting with them. And so, my office hours, like I gave my first exam last weekend, and so my office hours have been super full of students all the time, which is great.
An exam already?
Yeah. So I get into teaching philosophy and stuff, but I believe in having multiple exams so they're not high stakes. And so, we've got our first one done already. So, but yeah, interacting with students and just getting all the details and getting everybody's schedules done. And actually, part of my week this week has been the schedules go live to you guys to actually see in late February. So that means we have to turn them earlier than that. So it's been a lot of going to faculty and saying, what do you want to teach? When do you want to teach it? How many people can I put in a class? I'm also serve as the assistant department chair, so that's one of my jobs as well, is kind of organizing all of that.
Nice. So just to let you know, that water is yours.
Oh, thank you.
So if you need a drink, go right ahead. There's also a little snack there too. If sometimes we'll be like an hour in. I'm just like, oh my god, I'm starving and I can't even think straight. I'm like, I need something, man.
Oh goodness.
But okay, and so yeah, how's your week been?
I think it's always busy, but good busy. So I usually teach somewhere between 9 and 12 hours. That's my load usually. And
so what is that? 3 classes? 2 classes?
3 to 4 classes, depending on what I'm doing right now. I'm doing 9 hours right now. So because I have release time for research. So my week has been teaching and I have 4 amazing research students who are in and out of my lab all the time. And so it's been interacting with them, getting ready for Oklahoma Research Day, push for that. Abstracts are due today.
Today at 5, right?
Today at 5, yes. Yeah. If you want in the program, but you can still go even if you haven't submitted the abstract. So.
Yeah, I think that was one thing we were also wanting to talk about is, because I know you're a big advocate for research.
Yes.
And so maybe sometime later we can talk about how maybe why you think research is important and sort of how students like me and Albert can get involved.
Absolutely.
Yeah.
I will say when I first started out, when I was back in high school, I wanted to do something where everything aligns. I wanted all the numbers and all the things to be perfect. So I said, I'm going to be an accountant. I've got all of that going for me. Yeah. And then somewhere along that way, about my junior year of high school, I took my first chemistry class and I went, it's accounting with molecules. And it can't lie to you. They always have to
That's
add
a beautiful way to put it.
Right.
I like it.
And so I fell in love with it and started doing that. Went to undergrad as a chemist with a pharmacy kind of major in there as well. And I decided the very first month I was in undergrad, I was like, pharmacy's not for me. It's for some people and they're great that they could do it, but I was like, I couldn't stand there and interact all the time. I was like, that's not for me. But one of the reasons I love student organizations is because that's how it got me into research. My very first month of my freshman year, I went to a students affiliated with the American Chemical Society, which is a mouthful, we call it CFACS. It's like Chem Club. And so I went to that meeting and they had all the new professors talking that were at OU that semester. And I always knew I wanted to do something that was kind of natural but still help people, and I just didn't know what to call it. And Dr. Robert Chehevich gave me my first shot at this. He talked that night, and it was called Natural Products Chemistry, and I fell in love, and I was like, this is what I'm going to do. Yeah. And I'm a little sad to say that was 21 years ago, unfortunately. Wow. But I fell in love with it as a freshman, and I went up to him and I said, hey, I know I've heard nothing but you guys don't take undergrads because it's OU's a more graduate student focused campus. And they said, no, come talk to me tomorrow. And this is like Thursday night. So Friday morning, 7:30, I'm sitting outside of his office waiting on him because I had no idea he didn't get in until like 8:30. So I sat there for like an hour waiting on him. And he's like, oh, I was so excited. And I said, hey, I'll volunteer. I'll do anything. Will you take me? And he's like, yeah, absolutely. And I worked for him for 4 years, and it was amazing. And I fell in love with natural products doing that. So
that's awesome.
What I tell students when they're like, how do I get involved? I'm like, take your shot. It's hard to say no in person, if you're sitting there waiting. And so that started the journey for me. And I've done natural products in one form or another, like I said, for the last 20 years.
Awesome.
Wow.
So on that topic of OU, did you do your graduate as well at OU, or?
No, so my graduate is actually from a school of pharmacy in Mississippi, so University of Mississippi, Ole Miss.
Okay.
Yeah.
And so do you know, because me and I were both are thinking about going to graduate school at OU. Mm-hmm. And if Tej doesn't convince me to stay here at UCO, and is okay, so I know you turn in an application, but then how do I go about maybe going and getting a TA position or RA position? Do you know that?
Yeah, so it's actually funny, getting back to what you asked me what I've done this week. I've been helping students prep for grad school interviews.
Okay.
So we've got several from the department, and they've got interviews coming up, and what do we do if we get in? What do we do if we don't get in? All those conversations and backup
I should have been there.
It was some good conversations. But yeah, and I realized it's changed a little bit because back in the day, February 1st used to be like our hard deadline of when you would apply. And now I'm seeing dates as early as like November and December. So always check your dates, always check how that goes. As far as, I think the biggest thing students don't always know is that when you're in a STEM field, particularly engineering, chemistry, biology, we don't pay for our grad degrees. So we are either working for a professor or we're, like you said, TAing, being a teaching assistant, being a graduate assistant. And so really most of the time when you go to graduate school, the only thing at most you're paying is your fees.
Yeah.
And so that's really nice, and I think a lot of people don't know that, because I talk to so many students and they're like, oh, I don't want to go in debt for grad school. And I was like, you don't have to. It's okay.
So sorry to pause you there, but
Yeah,
Wow.
please.
So I didn't know that.
Are you saying that I should have applied in February 1st?
That's what the chem programs are. Check what your engineering programs are, because I'm not sure off the
from
top of
what
my head.
I saw, it was there was no due dates. I couldn't find any due dates for anything.
So a lot of, and like I said, I can only speak for the chem programs and the pharmacy type programs, but they are more December, January timeframes, that you're applying. Yeah. But engineering's a little bit of a different field. You guys are a little bit more applied than we are, so different kind of things going on.
Interesting. Okay.
Also, one other weird fact that people don't usually know, and thank you so much for a wonderful bio that you gave me, but you notice I don't have a master's. And so it's also very common in STEM fields that I go straight from a bachelor's to my PhD. We don't usually stop and get a master's. You can. I have other faculty that do it, but you are also welcome to go straight from bachelor's to PhD.
Yeah.
So I heard the difference is one is a thesis and one's a dissertation, right? So if you complete your research and you publish your dissertation, you're basically a doctorate.
There's a few more classes at different levels, so just like you would have an undergrad level would be your 1,000, 2,000, and your grad would be your 3,000, 4,000. Your master's is usually going to be 5,000, and your PhD would be 6,000. So you have to make sure you have enough 6,000 level classes.
I see.
Oh.
So you said you were doing research throughout your bachelor's. Now, so did you have a thesis ready whenever you were going to grad school?
Different projects. So I wrote a thesis and presented a thesis for my undergrad, for my bachelor's. Okay. So I did have a project and did publish a thesis for that. And then when I went to Ole Miss, I started on a new project, knew everything, and took the classes, the graduate classes, and then wrote my dissertation from there.
How did you because that's sort of what I'm a little worried about, right? Is because I love material science.
Yeah.
Like ever since I met Tej and he's convinced me to do some material science research. Well, he didn't have to convince me too hard, but
Not too much arm twisting.
yeah, but no, he showed me sort of the why material science is so interesting, and so I would like to keep doing. So my research is in machine learning applied to material science. So I would love to stay in that vein, right? But I'm not too. I don't know. I am also up for a challenge to switch it. So, do you think that was good for you to change directions, or like, would you have rather stayed in the lane that you were in, or?
So, a little bit of an involved story, but one that I recommend for everybody. So, during my academic years, I worked with Robert the whole time, and I did natural products, and that was my thing. I was isolating, I actually haven't said what natural products are, isolating basically pharmaceuticals from natural sources, be that plants, be that animals, be that fungi, bacteria, that type of stuff. Sponges are a fan. I kind of got into marine natural products, which is a lot of sponges over the years. I also did a lot of harmful algal bloom work over the years.
Oh yeah, I saw that paper. That's pretty cool.
Yeah, that's carbotoxin is one of my favorite molecules ever. Paid to work with, but amazing to actually get to publish on it. But I took my summers and I used them to go do other research opportunities. So my sophomore to my junior year, I had applied and was a Fleming Scholar at Oklahoma Medical Research Foundation, so at OMRF. And so I was doing much more biology, microbiology type of things, PCR, gene constructs, those types of things. And I love being in the lab, but I'm not a microbiologist. If I never see another, PCR again or, protein purification, I would be very happy. Yeah. So I knew that I could do lab things, but it was natural products was a special part in my heart, right? And so then the next summer between my junior and senior year, I was lucky enough and applied and was a NOAA. So around here we think of NOAA as the weather center, but really they do so much more stuff. They do all of the marine biology, they have natural products, they do all of your fish counts and stuff. It's really much more than weather. We just think of weather here because we have such a great center in Norman. But I was a NOAA hauling scholar. And so, which was a nice scholarship, which, is a great way to pay for college. But I got an internship down in Charleston, and I was working marine toxins, natural products research down in Charleston, South Carolina for the summer. And so it was like, okay, it's not just being with Robert in his lab that I loved, it's natural products, and I always loved doing that. And so that really, those different opportunities let me find myself as a researcher, as a scientist. Sometimes you have to do something you don't think you're going to like to know that what you like is best for you, right? Sometimes you need those projects. And so when I went and applied for my PhD and was getting my doctorates, I went, okay, I definitely want to do marine natural products. At that point, the universe was kind of shouting at me, harmful algal blooms, that type of thing. You can't ignore it. And so I went to Mississippi and worked with an advisor that did all of that natural product stuff. And the great thing about grad school when you pick a lab is some labs are very siloed and focused, and then other labs are kind of broader. So even though, yes, I was doing natural products as a whole, I was doing, bacteria producing certain anti-cancer compounds that also live in the sponge, so that's like an ecology project of the chicken or the egg, who came first, the bacteria or the sponge to produce the compound, and really fascinating ecology stories. I did some total synthesis. I'm not a total synthesis person, learned that early on in grad school, but I did it a little bit. So you get to explore and have different projects.
Could you briefly explain what total synthesis means?
Yes. Yeah. So as a chemist, particularly a natural products chemist, we produce these and find from nature these amazing molecules that sometimes are very, very large, very complex. It's always a great story that, especially with the AI right now, everybody's like, oh, computer generated, the computer can come up with all the structures you need. There are structures out there that nature makes that you couldn't even comprehend until you actually find it. And so there was a big push in pharmacy in the late 90s to kind of do away with the isolation and elucidation, which is just a fancy word for saying taking something from nature and finding the compounds in it. And they're like, oh, we can do it through computational chemistry and it'll be fine. Production went way, way down. They weren't having new molecules. I mean, you can look back and see the FDA, Food and Drug Administration, when you submit it.
Wow, so there is an actual trend when people shifted to computation and they couldn't get as much of an output.
Right. And then they shifted back again, and you can see it go up as you're going through it.
Interesting.
Wow. Yeah.
Interesting.
So,
that shows the limit.
Nature, amazing. It can do things we couldn't even comprehend. Yeah. And so total synthesis, you're not always going to get enough molecule from a natural source. Paclitaxel, blockbuster cancer drug. I mean, it's the first line of treatment for most cancers nowadays. You'll hear it called Taxol quite a lot. It comes actually from a Pacific yew tree, and those Pacific yew trees are slow growing, and so you don't want to be cutting down the forest basically to make enough drug to save people, but you still want to save people. And so what they do is they figure out, chemists, very talented chemists, figure out what reactions to put together to make this molecule without having to cut down a tree. Oh. And so the total synthesis is basically making it from scratch. Yeah.
Wow.
Now, in the case of Taxol, that's also another interesting story. You can get a precursor compound, like a compound that has a lot of the stuff in it but isn't the final compound, from the needles of the pine tree, the yew tree that falls. And so there's huge collections every year to get all the needles, because that's not going to hurt the tree. It's like collecting leaves. And so they take those yew trees, get the starting compound, and then modify it.
That's amazing. Okay, wait.
How do you spell this? Yew trees?
Yeah, that one.
Yeah, okay.
Well, it's the Pacific, not the European, but either way.
Oh, okay.
So
YEW, yeah.
These things right here?
Yeah.
Huh.
Wow, they look beautiful.
Oh, they look like the trees that you see in Hawaii.
Yeah.
I wonder if they look. It's got to be the same thing.
It's similar, yeah.
Yeah. Huh. Wow.
Well, okay, so I'm curious, because you've done so much research since undergrad, postdoc, and all that. Which is your favorite? Because you said you have discovered a few things that you like, a few things you don't like. So, which is your all-time favorite so far?
Like activity to do or project that I've worked on?
Project that you worked on.
I think it goes back to you spend so much time, it's a labor of love for your dissertation, and a large chunk of my dissertation comes out of carlitoxin. And so carlitoxins were these molecules, there's a series of them, that were discovered from Carlidinium vinificum, which is a harmful algal bloom. It's actually, you can think of like the green tide, the red tides, it'll cause those types of events. Those types of fish killing events. And so this was a compound that came out of those blooms, and it was really interesting because we took it as far as to actually turn it around and be able to target certain cancer cells. And so the last time, and I think it's still being worked on a little bit down, my advisor in Mississippi actually moved, weirdly enough, to Charleston, weird roundabout connection, but it's a small world.
Sure.
But they're still working on it a little bit for its anti-cancer potential. Yeah.
Wow. Interesting. So I want to step back just a little bit, right? So, we're always curious where people's curiosities come from, right?
Yeah.
Like it's something I spend a lot of time thinking about, right? Because I think until you find something you're extremely curious about, it's you don't really have the motivation to really do anything, right? You just like sort of wandering around, and but then once you find that thing that you're curious in, it's like an explosion in your mind. So where do you think maybe your curiosity comes from, and maybe what sparked it in this chemistry realm?
Yeah, because you said it was at first in numbers, right? But how do you, is it when you're in elementary school, when you're in middle school?
I was lucky enough to always be a really good student, so a lot of subjects came easy to me. So it's kind of almost deciding what do you want to do. It's harder to narrow it down when you can do a lot of different things at times. And I knew I wanted to help people, and coming from kind of a smaller town, and I had an amazing chemistry teacher who was also the physics teacher, who really was a bio major, small town living, awesome. and she was wonderful. I adored her, and she was like, well, I think because you like chemistry and you want to help people, that means you want to be a pharmacist. And that's what she knew, and that was great, and that's why I kind of started out with that. But as it kind of evolved, I realized my brain loves puzzles. I'm a puzzle person by far. I'm also a Legos person. Like, I love to see all of that come together. So anything where I can actually make all the pieces fit is always very satisfying to me. And so part of what I do in my research, I talked about isolation and elucidation. That elucidation part is, okay, I've got a vial in front of me. What is that compound? How did the carbons connect? How did the hydrogens get there? What's the oxygen doing? And so leading into the nuclear magnetic resonance, the NMR class that I teach, that is putting all those pieces together in a way that makes sense for the molecule, that then once I know what the molecule looks like, I can give it to my total synthesis people and they can make it. So it's all part of this together.
That's pretty wild.
Yeah, so my actual area of expertise is structural elucidation. So I'm an NMR person by training, and so you will give me an unknown compound that no one knows what it looks like in the world, and then I collect all of this data and have to put it together in puzzle pieces.
How many times have you done that, do you think?
Oh, I don't know anymore.
Is it like second nature to you now?
It is second nature, and it's one of those where it's a skill. You practice at it. When I teach that class, which I have amazing upper division chemistry students, and they look at it and go, there's no way we're getting the structure out of this. And I'm like, we will. We're going to work through it. It's going to all make sense. I introduce them to new techniques beyond what they see in organic chemistry, because there's like a one-dimensional NMR, and then there's a two-dimensional NMR, and we talk about crystal structures and how all these pieces of data come together to actually get those.
The structure. So NMR is just one step.
Just one step, yeah.
So like, what other steps are there? Crystallography or like...
If you're really lucky. So crystallography, everybody thinks, oh, I can get a crystal to grow from a compound. Not all compounds will crystallize, and not all compounds will crystallize well. And so the bigger the compound, the harder it is to crystallize. So like if you try to crystallize a protein, for instance, that's super hard in very specific conditions. That's what I'm trying to do.
That's why it's so expensive to crystallize proteins and all that.
Very, very much, right? It's a lot of trial and errors to get them when you're doing that. So, if you can get a crystal structure, it's like taking a little Polaroid and you get the data and you're like, oh, this is beautiful and it looks great. That maybe happens 10% of the time.
So are you having to use like an XRD machine to get the crystal? Or
if you did crystallography, you would use an XRD.
So, let's say we found a stubborn molecule that will never want to crystallize.
Then you do it in solution, and that's where the NMR comes in. So I'm able to, through a series of experiments, put this compound into solution. Usually it's chloroform, methanol, something like that. It's a deuterated solvent because it has to do with how the machine has to work. And so I do different experiments. One will show me how the protons are connected. One will show me how the carbons are connected. One will show me if it's near an oxygen or not, if it's near a nitrogen or not. And then there's even other experiments I can do once I know kind of the two-dimensional flat structure-ish. I can then do other experiments to say, oh, well, this hydrogen over here, on the far left side can see this hydrogen on the far right side, so they're...
Hello, space travelers. Want to look sharp while bending space-time? We've got brand new Physics Society t-shirts. The clean graphic tee is just $20, and if you want to drop some Einstein-level wisdom on the back with this quote, I have no special talents, I'm only passionately curious, that one's only $25. Rep your curiosity, rep the cosmos, and support the Physics Society. Grab yours today before entropy takes over.
Has to be some type of folding that's happening, and so I can even get more 3D information out of it.
Wow. Okay, wait. I'm just curious because physicists, at least because I used to be a very big astronomy nerd like him, so we usually think about identifying compounds using spectroscopy.
That's spectroscopy, yeah.
So you So do you guys use spectroscopy a lot?
Yes. So anytime I'm running NMR, that is going nuclear magnetic resonance. That's going to be actually nuclear magnetic resonance spectroscopy, and so that's all of that. And then also with that, you can get a lot of information with mass spectrometry, and so that's going to tell you like weights and things, and you can see like
Estimated molar mass.
Estimated molar mass, yeah. Like if I fragment a molecule and I break it in pieces, what do certain pieces weigh, and how does that go together?
So
all of this comes together to make a structure.
can you so
That blows my mind.
can you tell us how this thing works? How does this NMR?
Yeah, so when you have an NMR, which we do on campus, we have a 400 megahertz NMR, which is amazing. Love it.
Okay.
Even though it takes a lot of my time as well, because it is a superconducting magnet. Okay, so it has to be kept very, very cold. So it has an actual metal coil that has been energized to cause the actual magnetism to occur. In order to get zero resistance, you have to cool it to liquid helium temperatures. And so to keep the liquid helium, because that's really expensive, you actually fill it once a week with liquid nitrogen, and the liquid nitrogen keeps the liquid helium cool, and the liquid helium keeps the wire cool, essentially. So you've created a magnet, and when you put your sample into the magnet, all of your electrons are either going to align up or down. It's arbitrary, it's a 50-50, because our concept of north and south is based on the poles, but a molecule doesn't know that. So it just says half the electrons are up, half the electrons are down. And then you pulse it with a radio frequency pulse, and that knocks the electrons off the axis, and then you measure as they precess and come back up into position, and how long it takes them to come back up is what environment they're in.
So it's a tinier MRI.
It is exactly an MRI,
Wow.
except, so here's always the funny story I tell my students, it's exactly an MRI. But they found when they were developing MRIs that people didn't like to be spun around and around. So in an NMR, the sample spins. In an MRI, the machine spins so that the person doesn't have to.
Yeah,
but the technology is the same.
Dr. Bin Jabbar actually gave a talk in front of Physics Society last semester about how, not necessarily just how, what did you... It fell out of my brain. The machine you're talking about, MRI machine works, and like the history of it, like all the physics it took to get us to that point.
It was used for astronomy at first.
Yeah.
That's exactly what I was like alluding to. It's crazy.
Well, and I emphasize this in my spectroscopy class, because we've changed the name a little bit since theory and practice of NMR, because spectroscopy is a little more holistic. We include mass spec, crystallography, infrared, IR type of stuff as well. So we do it more holistically than what that original class was. But I always tell them, look, the physics, we owe all of the instrumentation and the design to them, and all the new advancements that we ever have in NMR techniques, because we can get stronger and stronger fields, we owe to them. The interpretation, that's ours.
Yeah.
And so you're not, and when they did the actual Nobel Prize, which people have won Nobel Prize for NMR, there were Nobel Prizes that were won for in physics for actually the development of it, and then in chemistry for the implementation.
Wow. Everything sort of points back to magnetism. So
In that regard, yeah, absolutely.
That's so crazy.
Yeah.
Wow. Yeah, because in our lab, that's our specialty is magnets, right? You synthesize magnets. A lot of the simulations I run is based off the Ising model, and so yeah, very interesting. That machine's got to be really expensive to keep working, right? Because you said you have to put in
liquid nitrogen and all that.
Yeah, liquid nitrogen's not nearly what you'd think. It's not too bad. You fill it once a week, we're good. Liquid helium can get into the thousands of dollars to fill. We fill 3-ish times a year, depending on how it goes. The actual machine itself, $250,000. So,
wow. Yeah.
I would be willing. I know it is the most expensive piece of equipment in the chemistry department. I'd be willing to bet it's close in the entire college. Yeah.
Yeah. Our most expensive is $100,000. Our XRD machine that we get is about $100,000. Wow. Could you take us maybe on a trip through this class, maybe just a summary of like what do you teach in this class?
Yes.
Like where do you start and where do you end?
Yeah, so like I said, we kind of changed the name up. It's now spectroscopy, structural elucidation with spectroscopy, or
That sounds so cool.
spectroscopy for structural elucidation, something like that. When I had a predecessor before I came here, and I was at UCO in 2016, so he had retired the year before that, and he was an amazing NMR person, but from the physics side of things. He could tell you how the instrument worked inside and out, and he could tell you how it all came together and all the physics behind it, and that was wonderful and very, very important to someone, not necessarily our students who are going out in the field and being asked to say, what's this compound? And so I took what was that NMR class and really developed it more into a problem solving. We still talk about the equipment, we still talk about the importance and the history and everything, but now it's, all right, remember in organic chemistry 1 and 2 where you learned about infrared spectroscopy, which is IR, and what the functional groups are, and then remember where we learned a little bit about mass spec and how, different, if you have a nitrogen, you're always going to have an even or an odd mass depending on how many nitrogens you have. Those little things, a chlorine is going to show up with a very specific pattern in a mass spectrum, those types of things. And then we get into 1D NMR, which is always silly to me. We call it 1D NMR, but it has 2 axes, so it's really 2D NMR. But anyway, that's a field thing that's always been like, why do we do this? Yeah. And so in organic chemistry 1 and 2, you learn how to read the proton NMR of it, so you can see all of the hydrogens. And they touch briefly on the carbons, but not much. So that's probably the first 3 weeks of class is me doing the review of what you've seen, and then I go, okay, now here's all the other really cool stuff that they didn't tell you. And so I'm like, how much easier is it to do a structure if I know all the carbons? Or there's different experiments where a carbon that has 2 hydrogens will appear face down, and a carbon that has 3 hydrogens will appear face up. And so I can tell the difference in them.
Wait, face up and face down?
When you're reading a spectra. Yeah, when you're reading a spectra, instead of having, keep hitting the mic, I'm sorry.
You're fine.
When you have a spectra, you've got a line going across, and so normally when you look at a spectra, like if you pulled up a proton spectra, you'd see all of the lines going up, but there's certain experiments where different carbons can show in different environments. So everything that's like a CH2 points down, everything that's a CH3 or CH would point up.
Is there something we can pull up to visualize this?
Yeah, absolutely. Do a depth spectra, DEPT.
Depth spectrum?
Yep. Spectra, yeah.
Spectra.
Oh, you might need to space that.
Yeah, there you go.
Oh,
and so if you click on that first one right there, it's showing you the differences of what they look like.
Let's see if it'll.
Yeah.
Okay.
Now that's helpful.
And so you can see the different environments and how they are.
And is this what you mean by flip?
Face down, yep.
Okay.
So from this, you can get a sort of 3-dimensional.
You can start to see how the carbons and hydrogens interact.
Okay.
And so this gives you more information, but the more information I have, the more I can do. So then we move into what's called 2D NMR. And so now the experiment isn't just detecting protons or carbons, it's detecting protons attached to carbons. And so I can see direct correlations between that. And then there's other more advanced experiments that let you see not 1 bond away, but 3 bonds away, or 4 bonds away. And so you start putting all those together. Yeah.
Huh.
Wow.
And so that class finishes with us doing 2D NMR.
I can see why you mean it's like a puzzle, because you deduce using these hints they give you.
Yes.
Yeah.
And all the data has to make sense. The thing I say so many times in that class, I was like, absence of proof is not proof. Just because you don't see it doesn't mean it's not there.
You need to falsify it.
Right, exactly. It just means you didn't pick it up in your experiment.
So why would you maybe not pick it up in the experiment?
So that goes back to the physics side of things, where in chemistry, something that's real in NMR, something that's really big is called a Karplus angle. And so a Karplus angle is telling you how Karplus is telling you where you are and how invisible you are to the pulse. And so if you end up being directly 90 degrees from it, you disappear completely. So you've got to be in a certain area where you can see it.
Interesting.
Wow, that's insane. insane.
Yeah, so just because you don't see it doesn't mean it's not there. It just means you have to tweak your experiment a little. And that gets more into the physics of, do I run it just a little bit longer or do I pulse a little bit harder with that RF frequency? Those types of things.
Okay, so yeah, what are all the parameters that you're sort of tweaking here?
A lot.
Okay.
So there's things in NMR that are called relaxation delays. And so you have to decide how long, when that molecule goes to its 90, when you've pulsed it, how long do you give it to come back up before you start reading?
Okay.
If you give it too long, you've missed the carbons that have a lot of hydrogen. If you give it too short, you miss the carbons that have no hydrogens. Because carbons that have no hydrogens are going to relax much, much slower than a carbon that has hydrogens attached.
Interesting.
And so all of that comes together, and so there's something called a T1 and a T2, different relaxation delays for different transfers. And so you play with those a little bit.
Yeah.
It's a lot, yeah.
That's amazing.
I've always been really, well, I wouldn't say that. Ever since I got into
STEM.
Into STEM and sort of going down this rabbit hole of physics, right? There's a lot of things that you read that say, oh, well, we can neglect the chemistry and all of this and that. And so I've sort of not necessarily looked at chemistry as a negative thing, but sort of just like, well, why do we need it? But then, just listening
To what you say. I'm
you
already
say,
sold.
there's a lot
chemistry.
of physics in chemistry, and I'm just
So
like,
much.
that's...
Yeah. Well, we have a whole class called physical chemistry, right? And so you have to have physics 1 in order to do that class.
Yeah.
That's amazing.
And I agree with you. I think sometimes in science, more so in the past, we've gotten very siloed of like the chemists do their thing, the physicists do their thing.
And I hate that.
Yeah, the mathematicians are over here doing something else. And then every once in a while you look up and go, man, I really need this equation or this figured out. And you go, oh, well, the math people did that 2 months ago. Great. And so you have to kind of keep your head on a swivel. I love the interdisciplinary approach.
No, that's why I've been telling him all the time. I said real world problems are interdisciplinary.
Always.
Always.
Yeah.
And
And that's why collaborations are so important. And you're kind of talking about where do you choose in grad school? Choose grad schools that have good collaborations, because there were times in grad school where I wanted to know how a certain test or something worked, and I would go live in the chemistry department for a month and hang out with them and figure out, oh, this is how we run this and this is how we do this, and then I'd come back to my lab. And so I would have these little like mini sabbaticals in different departments to figure out what I needed to know to make my research better.
yeah,
That's awesome.
I saw a lot of interviews from a series called SciShow Field something. But basically, they're interviewing a lot of PhD people about their research. A lot of them emphasize like collaborating with different labs.
Oh yeah.
Well, Dr.
Teo
just
says
gave
that
a
too.
talk over his research and how he's using sort of graphene to do DNA sequencing, right? He's doing the simulations of it. And he talks to us all the time about, the importance of trying to not necessarily reach out to people, but like sort of open your mind to being able to give part of a project to somebody else, because that, being able to collaborate can also allow you to get things done more quickly as well.
Absolutely. You don't survive. One of the biggest things when I came to a PUI, predominantly undergraduate institution, which UCO is, which I love the environment, but you don't survive trying to do everything yourself. My molecules or what I discover as compounds are nothing if I don't have bio data to back them up. So I do very little bio assays, like a test to say whether something is active or not, whether it kills cells or not, something like that. I do very little bioassay in my lab, but I partner with great collaborators in biology, and I'll say, hey, here's my stuff, tell me what it does. And it works for both of us. And then we're training multiple students, not only in the chemistry, but in the biology. And I'll send my students over to Dr. Vaughn is great. I love him. I've collaborated with him since I've been there.
I'm actually, he's my mentor right now.
I love Dr. Vaughn. And so he does, Dupuytren's contracture. He's got the cell lines and everything. And so he'll test, anytime he's testing an extract, it's testing our stuff.
Yeah, ever since I got into UCO like 2 years ago, I saw every time I look into research stuff, the moment that made me confident enough I want to like stay and do research here was when I saw CIBER.
Yeah.
Because I was like interdisciplinary biomedical research. I'm like, that's why I want it.
Yeah, you know, and you just keep reinforcing the reason why this was a great idea. Because so while after you said you would do this, right? So I would just go and tell everybody. Like ever since yesterday, I've just been telling everybody, like, hey, we're gonna have Dr. Waters on, and I run into quite a lot of people who know you.
Yeah, a lot of students were like, oh, we love Dr. Waters.
Yeah.
In my defense, I teach a lot of Gen Chem 1.
That's right.
So you don't usually get through Gen Chem 1 without me. Which I love, because then I know all my students. Just one of the things I always have to say, I love this about teaching. I do a 1000 level. I get them at the very, very beginning when you're little chem babies and don't know anything. And then I do my spectroscopy and my medicinal chemistry 3 and 4000. So I see them at the very end. And so it's just really amazing for me as an instructor to be saying, here they're starting out, they're going to make it. I know they're going to make it. I feel good about this. And then at the end, where I'm writing their rec letters for grad school or med school or pharmacy.
That's wholesome.
And I was like, this is so awesome. I mean, it keeps me going all the time.
Yeah.
I love that.
Yeah, no, and so what a few people have told me is that you've been a great mentor to them. You're a great professor, and so. Was that something you got from your the guy you were doing work with during your bachelor's, or was it maybe a family member that sort of?
So that's really interesting. So, because everybody asked, oh, you know, you must have known you wanted to teach. No, that was not in the cards for me in the beginning. I was a shyer person. I didn't, wasn't very extroverted. I loved what I'll call a lab rat. I love being in the lab. I'll spend all the time in the lab. I mean, we were, when I was an undergrad, we were working we had a large group of undergrads that worked in the same lab, and, we would be there till like 10, 11 o'clock at night, and finally our advisor would kick us out and say, look, you gotta go. You can't keep running this experiment anymore. He's like, I gotta sleep at some point. So, so I love that. And I just really, I'm not gonna say I didn't like people, but it wasn't, interacting wasn't necessarily my big thing. And so I was like, I'm always, teaching wasn't in the cards. And I got to grad school, and the same thing. I was a teaching assistant a little bit. My grad school was a little bit different because it's a pharmacy school, so actually when I taught PharmDs. I didn't teach like undergrads or anything like that, so it's a different group of students, different dynamics when you're teaching a professional set of students. And so I would mentor students here or there. I'd mentor a PharmD student. I'd mentor another graduate student, things like that. And then when I went to NIH, there's not a large population of undergrads, right? You're all PhDs, you're all postdocing, which is the step after you have your PhD. You spend a few years doing postdoctoral work. And then I had a few PhDs hanging around, and I realized I missed it. I missed the interactions, and I missed, involving other people in my research and explaining what's going on and things like that. So the opportunity came up to come back to Oklahoma. They happened to be looking for an NMR spectroscopist, which I was like, well, that's me. And
you got it.
I was like, I knew I never ever, and this is me personally, I never ever wanted to be at an R1. That just wasn't my style. I didn't really care for the publish or perish world that an R1, a research intensive institution, lives at. So I had always seen myself government lab, maybe industry, although pharmaceutical industries, you almost have to put your ethics behind a little bit, which is always hard choices for me. So I thought I'll come back here, give it a shot. Well, first, I have the most direct career path anybody could possibly have, which is really weird. And everybody always says, well, how did you do it? How did you get to where you are? And I was like, I am super atypical. Do not do what I do. It was a weird thing. So I applied to one college. I applied to 2 graduate schools, turned down the interview with the second one because I got into the first one I wanted, so I didn't even bother. I applied to 1 postdoc position, which is unheard of in that climate, and got it. And then I applied to 1 faculty job in my entire life, which never happened. So like I have a very linear of where I was going, and that's very atypical.
Well, it almost sounds like people could see the potential in you.
So, I mean, getting hired was kind of a funny story because I was I was doing a group of postdocs, and you can only, interesting fact, you can only spend 5 years at the NIH as a postdoc before they push you out, because they don't want what they lovingly call permadocs. They don't want you staying forever.
Okay.
So you get a 5-year timeline when you're at NIH.
Interesting.
Because I have friends that spend a decade as a postdoc in other places, which is not great for your career. And so I was doing a group, and the group meeting was, how do you apply for jobs? And I was like, I'll go out and look, and I'll bring back stuff to the group of like, hey, try this website or this website. And I looked, and I was like, let me just see what Oklahoma has, because I was in Maryland at the time. And there was a job in Oklahoma, like I said, in a Mars spectroscopist. I thought, well, I've never applied. I'll do it for experience. You got to apply sometime. Not looking to leave. I was only at like a year and a half at NIH at that point. And I told my boss what I was doing. He's like, yeah, good experience. And I was like, they're looking for a chemist. Technically, my PhD's in pharmacy. They're not going to worry about me. And I got a Zoom interview. Actually, this is how old it was. I got a Skype interview.
Okay.
Oh, wow. I know, right? Yeah. Back in the day. It was 2015, but still, we were doing Skype back then. So they interviewed me, and I was like, it went well, but, they're not going to invite me for an on-campus interview. That's not going to be a thing. And I get a call the next week, and they said, hey, can you come on campus? And I went, okay. So I went back and told my boss, I'm like, hey, can I have, 2 days off to go fly to Oklahoma, do this interview, and come back? And he's like, yeah. And I'm like, look, I'm not serious. I'm not going to take it. I've never done it before. I want experience, on-campus interviews. And he's like, at this point. Which I had an amazing advisor at NIH. He was wonderful. I loved Kirk to death. And I came out here, I interviewed, and it was the week before Thanksgiving, so I was actually flying back here for Thanksgiving with my family. And I got a call from the dean's secretary saying, hey, will you talk to the dean? And he was like, we're going to offer it to you. And I said, what? I said, I have no teaching experience. I was like, I taught as a TA. And they're like, no, we're offering it to you. And I said, okay. And so then I went back to my boss at NIH, and I said, you're never going to guess. And he said, they offered it to you. And I said, I don't know how. I don't know why. So anyway, I said yes, which I'm so glad I did. And it was a hard decision. Sometimes you have those kind of life-changing decisions of, do I leave the bench? Do I stop doing, I don't stop completely, but do I back off of the hardcore research that I've been doing to pursue this opportunity? And I thought, I can do anything for a couple of years. We'll see how it goes. And turns out I love you guys, and I love teaching, and it is amazing. And I couldn't think of not doing it. Like I said, your successes, I like to share a little part of all of your successes. So to me, it's so much better than if I'd stayed at NIH and found maybe one blockbuster drug. I'm out here. I've got students that I've gotten old enough now that they're actually practicing MDs.
Oh, that's awesome.
I've had some go through grad school. I've had some of them go through grad school and come back and teach for us at this point, which is amazing. So I kind of switched what I thought my focus was going to be, which was, research to train the next generations of researchers, which I love.
That's the beautiful. Go ahead.
Yeah, if you don't mind me asking, is that so? You said that it's not that good for a career if you stay in a lab for like 10 years or something. So would you mind explaining why is a permadoc so bad for a research career?
A couple of things. One, the pay is not great. So you don't want to be at that pay level for quite a while. You're not getting promotions or anything like that. So it's pretty stagnant.
Unless you jump.
Right? So you really want to go. Ideally, postdoc is an extension of your graduate education, of your PhD or your master's, but usually it's your graduate education. And so it's a different lab with different experiences, getting trained in a few different techniques, I was still doing natural products, but I was now doing a little bit different natural products with different bioassays and things like that. Was able to work on, some really niche cancers that you wouldn't necessarily see in academia because there's not like the funding to go after those things, but NIH had it. And so you do different trainings there, and you just don't want to stay too too long, because you're not advancing your career at a certain point. It's good to move on to a research scientist. That's an amazing jump from a postdoc. Looking for a faculty line, that's always great as well. Going into industry, that's good. But you don't want to stay, they're not meant to be permanent positions. They're meant to be 3 to 5 year positions.
Interesting, because I've always imagined as a child, like, becoming a researcher, you just sort of find your lab, and once you find it, you just settle there, like, that's how I imagine it, but...
Not in this funding economy, you don't.
That makes sense, honestly, yeah.
Interesting. So what was, like, the interview process of going to the NIH, or applying for NIH?
I'm laughing a little bit here because I wasn't really aware I was interviewing when I was interviewing, that makes sense. And again, I will, a shameless plug for students going to meetings, because they are the best thing ever. You go to meetings, you meet other people from other labs, you get to see interesting research, but you're also making those connections.
Are we talking about conferences?
Scientific meetings.
Oh, okay.
For chemistry, there's large ACS meetings. Oklahoma Research Day is great as well, a little bit on a smaller scale. There's something in the world that if you ever get a chance to go to, take it. They're called Gordon Research Conferences. Gordons are amazing because normally your big conferences are going to be like 10,000 people, and so it's hard to really have meaningful conversations. But Gordons are usually 200 or less, and so it's just your field. Like there is a marine natural products Gordon conference.
Lipidomics.
Yeah, that's
ionic cluster. So they're very
niche. Yes.
Oh, cool.
And so you meet your people.
Yeah, that's what the MMM conference was for us.
Yeah.
Is magnetics conference, and we would just go and nerd out about magnets for a week. We went to Palm Beach last semester, and then last January we went to New Orleans.
Yeah.
And those to me, conferences was sort of the thing that like flipped a switch in me, and because one of the reasons why we started this whole thing, right, is that me and Albert get together, we just start nerding out, and we were like, man, we want to like create a space to nerd out with other people as well. Yeah. And
find our own people.
Yeah,
it's all
exactly.
about that.
And a conference is that times 10.
It is.
And so especially if you're presenting, let's say like a poster, right?
Yeah.
You have a bunch of people coming around, asking you a bunch of questions, and you just get that chance to like talk about the thing that you're most interested in and you're working on.
And when you're in the field for a while, they, like you said, they become your people. They're people that you've either worked with directly or indirectly, or you've communicated with and say, hey, I can't get this particular experiment to work. Do you have ideas? I saw you publish something similar. Those types of conversations. I've been to some meetings where it's like coming home, like you can't make it to check into the hotel because you keep running into people in the lobby that you're like, oh, I haven't seen you in forever. But so I went to one of the Gordon conferences, which I'd done for a few years, and with my grad work, and I was presenting my poster. I was talking to my actual advisor at Ole Miss, and he's like, hey, so-and-so, it was Kirk at that point, Kirk's gonna come by and look at your poster. He's looking for postdocs. And I was like, oh, I'd really love that. That would be great. It's a big conference. I'm sure he's, there's a lot of us here that are looking for postdocs. And Kirk came by, we talked, had a good time. The Gordon conference is much more relaxed than most conference. Everybody's over 21. There's an open bar.
Nice.
So there's a lot of talking around things like that. And he was like, do the application, send in the application, send me your references, and we'll talk. And I was like, okay, great. So this was in California. I fly back to Mississippi, and I'm getting my references. And again, it's a small world. My chair of my department, who was not my advisor, but it was chair of my department, personally knew Kirk really, really well. They'd been friends for a long time. And I said, hey, will you write me a reference? I didn't know that when I asked him to write me a reference. But I said, hey, will you write me a reference? And he was like, oh, yeah, no worries. And he's like, have you talked to Kirk? And I was like, yeah, I talked to Kirk. He seems, like this is a shot. I said, I really want this. I think it'd be great. And so I go back to my lab, and I'm doing research. And the chair of the department calls me, like, 30 minutes later, and he said, hey, will you come back? And I said, okay, yeah, sure. So I come back into my office. I said, hey, what did you need? And he goes, I just got off the phone with Kirk. I said, you called him? I said, I asked you for a letter, not to call the man.
Yeah.
And he goes, no, it's Kirk. I called him. And he said, I think you're under a misinformation. He's like, you're not applying like you're going into a pool and he's going to pick somebody. He's like, he's just doing the paperwork so he can hire you. He just needs to cover his bases and make sure you have a reference and everything.
Wow.
And so I was like, oh, and he's like, you're going to Bethesda. Like, if you want it, you're going. So it was really weird.
So then, okay, so you and this Kirk guy must have sat at your poster and you explained to him this, what you were working on. What exactly was that?
That was the carlotoxins.
Okay.
That was about 6 months before I wrote my dissertation.
Your core memory.
It's core memories, yes. You spend a lot of time with your work when you're in undergrad for that, or in grad school for that. So you have those projects that you dream about them because you're living them day in and day out. I mean, it's a 5-year project. So,
yeah. Wow. Wow. So
Okay.
Go ahead.
Okay. So, for us undergrads and future, hopefully, graduates and postgrads, how do we plan a project? How do we find a project that is considered like good for us or good for our career? Because especially in today's economy where grants look for impact or look for keywords, like for example in neuroscience they'll look for Alzheimer's, if not they won't accept you for some reason. So what do you how do we find some balance in between what we want to do as a freedom but also what
will get you funded? And that's a hard question. So it depends on the lab you go into,
also
for
depends
sure.
on the field too.
It feels on the field. It depends on the lab. So there were running projects when I went to grad school of like one of them happened to be, it's a molecule called manzamine A, and that was always my advisor's like pet project was manzamine A, and it was actually better than I'm forgetting the name of the malaria drug, but it was better than one of the current malaria drugs on the market right now. And so it was, artemisinin. It was better than artemisinin. And it was really good. It just was having some solubility issues, but he'd been working on that for a long time. So several of us, I even had my hand in it at one point. It was a small project that I was doing, but those types of things kind of exist through the lab, and it's kind of the next evolution. So you're really working for your boss. You're working for your mentor at a certain point.
So they already know.
Yeah, they have ideas. You can always take it in your own direction, but carlotoxin had been started 2 years before I got there, because it was, when I say it's a big molecule, it is like 63 carbons, 120 some hydrogens, 26 stereocenters, meaning they're either going to be carbons, the hydrogens are going to be one way or the other, and the stereochemistry really matters when it starts talking about the 3D shape.
Macromolecules.
Yeah, and so whether it's going to fit into a binding pocket of an enzyme or not, based on those types of things. And so they'd just done a big push and characterized the first molecule. And my work really followed up with the next 10 of them. And once I had a starting place, we were just kind of pushing our way through all the different variations. And some variations were better for anti-cancer potential than others. So then that kind of turned into a structural activity relationship study. You never know where a project's going to go. You got to be open and go with the flow a little bit of it. But the lab usually has like a central theme or a central idea that they want to go with, and you kind of fit in over your mentor's overarching one.
That makes sense. It's like it's like what you already want to do, which is machine learning, and then Tej already has magnetic materials. He tells you to.
Yeah. For me,
that makes sense. That makes sense.
Yeah, and my experience is probably a little unique, right? Because my first research advisor, Dr. Lamachani, he was just like, "Well, what are you interested in?" And I said, "Machine learning." And he said, "Okay, well, I'll find you a project that melds these, what I'm trying to do and what you're trying to do, and put them together." And so I guess like maybe the next step, but I have to find somebody who's doing machine learning stuff and then sort of in a field that I would want to go into. But that's what's been sort of nice about my trajectory is I just sort of, landed just somewhere perfect for me. Just serendipitously.
Yeah. So much so.
Yeah.
And I think sometimes the hard next step is when you set up your own lab. So I run a research lab here, is how do you have your own work that is not a duplication or an extension of where you went to grad school or postdoc. And so that's kind of that next step that nobody warned you about of, okay, if you're going to be a researcher on your own, you got to set up your thing. You can't duplicate everybody else's.
I also heard that that's also why postdocs are always buried in literature.
Oh, yeah.
Because that's how you know you're up to date, how you're not accidentally republishing someone else's.
So yeah, let's talk a little bit about that. So I guess first let's talk about like whenever you got here, what was the first thing you felt like you had to do, or did somebody give you like instructions
in
or
research or like just faculty life?
Research, because you're saying because you saying you had to build a lab, right? Like,
so I had a lab from scratch, and so I had the connections in Oklahoma as well, and so I one of my first emails and subsequent calls was to Robert Shahevich down at OU. He's since moved on to Michigan, but he was still there when I came back, and I said, hey, I'm coming back to take this position. I know you have this collection. I know you are not actively working on it and publishing on it anymore. Can I work out a material transfer agreement is what they're called, an MTA, and can I take it? And it was the samples and stuff that I had worked on as an undergrad. And he's like, oh yeah, they're just sitting here taking up space in my walk-in. So come down and grab them. You can have them, take them up. And it was a massive sponge collection that wasn't actually started by him. It was started by his predecessor, who was like a pioneer in natural products, doing marine natural products in Oklahoma of all places. Had one of the largest sponge collections. He's and his name was Francis Smith. He's since passed, but one of the deals when I took it, that both Robert and I wrote into the material transfer agreement, is if we find anything, any proceeds we have go back to the family, and I'm more than happy to do that when we find stuff, and give them first rights at do you want to publish or patent, things like that. And so he was great and amazing, and his sponge collection is from like the 70s, 80s, and 90s in places that you can't even go anymore because of political situations and in climates that don't exist anymore because of global warming and things like that. So like we've got stuff from Indonesia, and there's something called the Diversity Triangle, and so we've got stuff from all around that area. It was wild west of natural product stuff.
Before you move on any further, can you sort of explain like what is a sponge and why is it interesting? I know you sort of touched on it earlier.
So this particular collection, and we call it the Francis Smith collection because we named it in honor of him, includes sponges, includes seashell-ish type things, some corals, some different things. He would collect anything when he was diving, and so it was just kind of a little bit of a hodgepodge, which nowadays not legal. We have diversity things, but they came in before that, so they don't count.
Grandfathered in.
We're okay. We're not breaking any international laws. You would be today, but you're not breaking any of those.
Nice.
But a sea sponge, essentially, is an animal, which people never think it is, but it is an animal.
Okay.
It is an invertebrate, so it has no spine, it has no feelings, it has no central nervous system, okay? And essentially it's a sessile animal that just where it grows, where it's put down roots, that's where it stays the rest of its life. And they will actually spawn. There are times where I'm not making this up, the moon has to be just right, and you have to be in the right temperature of the water, and the sponges will put out their eggs and sperm and make new sponges, and then those will fall to the bottom, and that's where the new sponges grow. That's just how they work. But they are animals, and so because they are invertebrate, because they are sessile, they don't fall under any of the IRB animal protocols that we have to do. So they don't have a central nervous system. We're okay on that as well.
Is there like an interesting sponge we could look up, or should I just search sponge?
Just do marine sponge, yeah.
Okay.
I've got a canther. I can never say it right. A canther something on my head, but yeah, just do marine sponge. Animal. There you go.
Okay.
They're cute.
Yeah.
Yeah.
And so basically they will grow back. So you take a
Small
machete essentially and you're whacking them off. And then they grow back a little bit over years, and then you don't collect there for 4 or 5 years, and then the next collection trip you'll do it there. And so they are like filters essentially. You can think about them like the HEPA filter of the ocean. And they're just going to sit there and grow bacteria. They're bioreactors. Fish will come and munch on them. That's why a lot of them, they obviously can't move to get away from the fish, so they produce really interesting compounds to not be eaten.
So they are part of coral life.
They're not corals, but they are part of that undersea life, yes.
So pardon me. They are part of the reefs.
They can be. You can have deep sea sponges though. You type in barrel sponge actually.
Okay.
There's these things, barrel sponges are the biggest of them, and these things get as big as trucks.
Barrel. Wow,
Yeah, giant barrel sponge, yeah.
Oh, giant.
Yeah.
Oh, it appeared just now. Yeah.
Yeah.
Okay.
These guys are massive.
Oh my goodness,
Yeah.
these things! Wow.
Yeah, they're bigger than
huge.
people.
It looks like it's about to eat him, like it's a fish.
No, that's just his opening, but yeah.
Okay. So. Can you briefly
got a little
explain
face on it.
the anatomy of it? Like most sponges, I noticed they're either a ball of like a lot of surface area,
Yeah.
or they are like cones or barrels.
You need that surface area because for the most part they're acting as bioreactors. They all, I don't think there's any exceptions. They all have a very thriving microbiome with them. So whether that's sometimes fungi, not nearly as common, but lots of bacteria. And those bacteria, this is, I kind of alluded to this chicken or the egg story. Those bacteria are mutualistic with them. The sponge gives the bacteria a place to live. The bacteria will produce a compound that'll tell the fish not to eat it. And so they have this relationship that goes back and forth. Of it.
Huh. So it's sort of like making it spicy, and so something
Exactly. It's like, oh, it tastes bitter, I don't want to eat that. And then some sponges will see another sponge make something and like, oh, that sponge is red, so the fish know not to eat it. I don't make that compound, but I'm also red, so fish won't eat me either. So like the monarch butterfly type of
mimicry.
thing. Yeah, mimicry.
Wow.
Yeah.
So for something that doesn't have a central nervous system, the evolution is fast.
So, like, is there, like, how would I say this? So, is there different types of sponge,
Yes.
and then those have different types of bacteria growing in them?
Yeah, it depends on the location. So what you're seeing and what you're thinking of typically, because you were kind of SpongeBob.
Yeah.
You should remember SpongeBob's actually an animal, but you think of well, I mean, even your kitchen sponge actually is an animal at a certain point. Like if it's on a synthetic sponge, if it's a natural sponge, it's an animal. It's an invertebrate. It's been chopped out.
Wow.
So we're using a carcass to wash.
I mean, I'm pretty sure the ones I buy are just like
Probably artificially, but yeah.
Most of it's
I usually say artificial.
There's somebody out there. There's somebody out there with natural sea sponges.
The rich,
for sure.
rich people.
Exactly. But it depends on their location. You're thinking of the ones that are more tropical, but I've done expeditions where we're pulling them out of the Arctic Circle, and so there are cold water, like Arctic cold water marine sponges, which have very different bacterial communities than
So
something
have
that's
you
hot.
dived yourself into looking for the sponges?
I am always the boat person. I don't love to dive. We had a class in grad school that was research diving, which is very different than regular diving, and I didn't do it, but I'm the boat person or the lab person.
Yeah.
I don't mind processing everything they bring back, but I was like, I don't really want to be in the water. I didn't want to be stuck on a boat in the Aleutian Islands for 2 months. One of our lab mates did that, so.
So
So you have deep sea fear?
No, I just,
Not really.
I'm great with doing this stuff in the lab. That's kind of low-key natural products tends to be the Indiana Jones of the chemistry world. We're the ones in the forest in the Amazon digging up a plant. We're the ones diving. We're the ones running to places.
So this leads to a question that we were thinking about yesterday, right? And so do you think about like, you know, in this trying to discover new pharmaceutical drugs, right? Do you think about where at in the world would be like the next discovery? I know a lot of people talk about the Amazon, but like, are you specifically thinking about the certain like climates and ecologies going on? Like,
absolutely. I mean, you're always thinking about that. Funny story in the Amazon. Again, highly illegal. Before we had international laws to protect environments and things, there was a researcher who I will not name out of Arizona that went to the Amazon and literally by 55-gallon drums of dirt and plant material and everything and just stuck it in a warehouse because he knew that he wanted to study it one day. And some of that's still there. Again, not ethically responsible when you're doing that. You know, you want to not harm what you're taking. You know, with sponges, they grow back. It's okay. You know, you don't want to disturb a community or take the last of an endangered species. There's a whole area of natural products that does endangered plants, and so you always have to be really mindful that you're putting back as much as you're getting. But as far as where do you look, it's so fascinating because it's an organism. So let's say I was doing a project a few years ago on dandelions, okay? So I get a dandelion and I'm like, oh, this is a great compound from it. This is really interesting. I go back, I pick that exact same dandelion the next season, can't find that compound. The difference is 1 year we were in drought, 1 year we weren't. It's going to produce different compounds based on the different environments that it is. There's a study called ethnobotany, which is a really fascinating field. If you do more terrestrial natural products, which is land-based plants and stuff. So ethnobotany basically goes through and looks at any of the traditions, be that traditional Chinese medicine, be it Ayurvedic medicine, be it Native American lore, medical lore, and says, hey, they use this plant for this, we should go explore that and see what we get. So I have another collaboration that's coming up. We're going to start it this fall, also with the biology department. And we're
Dr. Vaughn and all that?
No, Vaughn isn't in this one. This is... oh, I gotta remember names. Two lovely ladies, Dr. Saxena out of biology and Dr. Messick out of biology. And so they are looking at traditional Native American plants, and they've already taken some of the extracts and know that they have activity against MRSA. I think they're also looking for
MRSA.
Yes, MRSA, and they're also looking for 2 or 3 other invasive hospital-acquired infections, and so they're looking at those, but they want to know what the actual compound is. So I've got a student writing an RCSA right now to do the isolation and elucidation on those, and I've got another student that's writing an RCSA to do the biology stuff for that.
Cool.
So, it's exciting.
Yes.
Yeah, that's going to start this fall.
Yeah, because like, okay, so you know, as a machine learnist, right? Like, I was just thinking, like, I wonder if there's like, you could, you know, somehow collect a certain parameters, right, from. And then sort of classify where at in the world would be the best place to go next, right? But like you said, like these things could change yearly.
It could be in your backyard.
Yeah.
You just never know. When you're dealing with nature, it's not always the same production every time. So you do have to kind of think about that.
Especially, okay, what are your thoughts on this? Because I saw this is a weird thing. I think it's out in Sweden or Switzerland. They have a storage of atmosphere from the past. So they tested it and they say that the air now versus 30 years ago smells different to animals and a lot of like
different oxygen content.
Yeah, different, like because of industry and all that. Do you think that with such rapid industrialization all over the world, pollution, climate change, we are and also the loss of diversity, we're actually missing out so much potential compounds that we can't research anymore. And do you think that we can still go out to these international places to like get more samples?
Right. I think that's why total synthesis is so important, right? So I've got sponges in a freezer in my lab from 1977, but I'm not going to find that sponge again. That country doesn't exist anymore on the geopolitical scale. Two, I'm not going to get permission to dive there again, not like you could in the 70s. So what's going to happen is if I find something great, I'm going to have to make it from scratch. I'm going to have to do that synthesis.
That's what you love to do?
Yeah, that's what you have to learn to do it. That's a harder question as far as, do you feel that pollution and other environmental changes are bad? One thing I haven't really talked about because it's a little bit detailed, but the pharmaceuticals we have, penicillin, any antibiotics, Taxol, any of those are what are known as secondary metabolites from plants and animals and
fungi.
Everything. Yeah, they're secondary metabolites. By definition, a secondary metabolite is anything you don't absolutely need to survive. So it's not your sugars, it's not your proteins, it's not your amino acids. Yeah, it's just extra stuff. You're not going to produce extra stuff. No organism is going to produce extra stuff unless it's pushed. And so it's challenged in its environment, or it has another species invading in on it. That's when you get the best secondary metabolites. So when I'm doing bacteria work, when I've done it in the past, I don't do it here as much, but I can get the same bacteria to produce different compounds depending on who I pit it against. Like I can pit it against another bacteria in the same dish, and it'll produce different compounds to fight off that other bacteria.
That's
so
cool.
like it's almost like a microbial fight club, which is really what it is. But that's the most interesting part of it. So you need some of that stress to get it to go.
Wow. So in a weird way, we are also allowing natural selection to make newer compounds in a weird way.
Absolutely, yeah. And so the decreased biodiversity isn't helping us, but everything else that's stressing us out is.
So I know that like they've talked about creating automated materials labs. Mm-hmm. Are they thinking of the same thing, doing that as well for chemistry?
To speed up research is what they say.
Yeah, so I can understand computer-assisted, robot-assisted extractions. Mm-hmm. That's manual labor for the most part. My poor freshmen know that. They do it enough. But there is an art to a separation to get the compounds, because I'll say, oh, you've separated the chemical goodness. So we take an extract, we soak it in ethanol, methanol mixture 3 different times. We get all of these compounds. It looks like tar, basically, but it's, thousands, 10,000s of compounds together in a jar, and we see if it has activity, see if it's kills certain cell lines, kills HeLa, something like that, and then we start separating it. And not every sample separates the same way every time. So there is an art to saying, oh well, I should use this stationary phase this time, I should use this stationary phase this time. You're tweaking things as you're going. So robots can do some of that. Mm-hmm. There's some planning that goes into it, but it's not as straightforward as it seems at times.
Yeah.
And then the elucidation side of it, they have tried for probably since AI originated back, even I remember people trying to do this even when I was in undergrad, so back in 2005, where they would try to take the NMR structure, the proton and the carbon and everything, feed it into a computer program and try to get the computer program to spit out a structure. Yeah. It can't. It can get some things, but there is an art that backs up how to put all the pieces together that no AI has been able to do. And unless it has come along in the last 3 months, I am not aware of any structural elucidation program that can do everything.
Have you heard of this thing called Claudebot?
Yeah.
Okay, so, it's this AI agent, and
I knew
I've been talking about this ever since, because, it's so... let's see. I think it's been going on for about a week now. I think they released it like a week, maybe a little more than a week ago, right? And there's already so much that has come from this, right? So essentially you can run it, it's a local agent, right? So there's like local LLMs that you can run on your machine at home or whatever, and then now they have a local agent that you can run. So you don't have to, go into ChatGPT and use the agent mode. You can run this thing locally, and it can do things on your computer, and then call like an API to an LLM to do sort of the thinking process. This thing just does the thing that the LLM tells it to do, essentially, the way that I understand it. And so, these things have been able to build some very interesting programs, and then also, they've created their own social media, which then they went and created their own religion and like all types of stuff, right? But where I'm alluding this to is, you said there's like a feedback loop that needs to happen, right? And us humans are really good in that feedback loop because we have all of this sort of creativity that we've built over time by doing this research, right? And we sort of know like what are the physical limitations, and I just trying to think of the far future, right? Like it seems like maybe it may get there, though, right? I'm not trying to take humans out of the loop. It's just...
I don't think we ever will.
So it goes back to, though, when we let computational chemistry try to create the most exotic molecules it could, it still couldn't do what nature did.
Because
so I guess
not in the data.
Yeah, AI is not going to think about it because it doesn't have anything to compare it to. It's going to, in my opinion, I think it would miss it because it's never seen it before. Why would it think that could happen?
Yeah.
It would think it's breaking the rules of its programming to try and do something that off the wall.
It's true. It's not found in its data set if it's too exotic relative to its data set.
Yeah. No. It's just to me, right?
Yeah.
I just think that, we're just thinking about right now, right? The capabilities of it, right? And it seems like the capabilities just keep getting better and better and better. And then there, starts to be... like right now, we're talking. The models are very. They don't have a good idea of the real world, but people are talking about maybe changing up what the model is, right, and giving it more of this physical, giving it more grounding
Spatial
in the physical reality.
Yeah.
But I don't know. At the same time, though, it's like then what do us humans do? If everybody's afraid, like, it's going to take all of our jobs and things like that. I'm just wondering if you've thought about this, because I know you talked about machine learning and computation earlier.
I go back to when ChatGPT first got released into the wilds, and how the free version, where it started learning from everything that was out there, and it actually became much worse. Like it couldn't function. And so at a certain point, you're only good as your learning set, right? As your data set.
Yeah.
So I don't know. It really scares me what data set AI uses to learn at times. If you give it the real world, I think there's so many exceptions, I don't know that it can deal with it. I'm not anti-AI, believe me. I actually just, thinking about all the random things I do in a week, had a conversation with a group of faculty about, well, where are we going with AI in teaching, and when do you have to disclose that you used it or that you didn't, or can AI actually grade for you? Is it competent enough to do that? Those types of questions. And so I don't know, there's a lot coming down the pipeline. I never want to say never about anything because we're scientists. It's just we just don't know it can do it yet, but it worries me, and sometimes we lose the creativity.
So something I've been thinking a lot about lately is, AI helps me a lot in not just learning, but also helping me produce little bits of code that I need and things like that.
That's great.
And so I'm just like, well, should we start giving it credit?
Yes.
Credit.
Right?
Because somebody invented that AI.
Yeah.
So that's been there was a big controversy that came out when we were doing scientific literature when AI started doing it, because journals, particularly physics has the same type of system, but we have the American Chemical Society journals or the Royal Society of Chemistry journals, and they have specific guidelines of, if you used AI to write your introduction, you have to declare it, and you have to say this section was AI generated, because the fear is you're going to start generating data that way, and that's not real data, and that's loss of scientific integrity.
Yeah.
And so they have come out with some clear guidelines of, if you checked it with Grammarly, whatever, you don't have to cite that. But if you put in this if you put in these 6 papers and said, write me an introduction, you have to cite that, and those 6 papers that you put into it, what you fed into it to do it.
So then because, I do this quite often, actually. I'll write, like just the other day, right? I was writing an abstract for our mechatronics project, and I put my abstract into ChatGPT and said, what do you think about this? And he said, well, it said, well, it's too emotional. and so yes, I understand, and maybe this is my rub with also like research papers in general, is it's just like very dry and to the point, right? Like for instance with textbooks, I like a textbook that has like some personality in it, right? Whereas you don't really get that when there's a bunch of people writing a textbook. It's just very dry and to the point. But yeah, so what about that though, right? Whenever I write the abstract and then it just goes in and modifies it, is that the same thing then, or?
It depends on how much it's modifying. So there's I haven't seen it published yet, but one of my colleagues took basically, I don't want to say tested us, but essentially took so many actual student paper abstracts and took so many AI generated abstracts and said, there was about 5 of us, and said, okay, tell me who was what, whether this was completely AI generated or whether this was AI assisted or if this was completely student work, just to see how the human eye could do it.
Yeah.
And it was interesting because what happens, and I see this all the time in students turning in this type of work, there is a consistent theme that will show up over and over and over again. AI is not good about varying what it's going to tell you. So there's a particular lab that freshman students have to write an abstract for, and basically you have a graduated cylinder, you fill it with water, you drop in a chunk of metal, and you find the volume of the metal by displacement. Like, that's the whole point. And I can always catch the ones that use AI because they say, oh, it's Bernoulli's principle. It's like, I didn't say anything about Bernoulli. Yes, it is Bernoulli's principle, but I didn't say anything about it. Yeah. You're a freshman. You don't know that. Yeah. I ask you aside in class, what's Bernoulli's principle? And you go, what are you talking about? Well, you put it in your abstract. Yeah. So, like, I think I don't necessarily have a problem with it if it is been outlined accordingly. So like for that particular assignment, because I'm trying to get students to learn how to write an abstract, I know AI can write an abstract. I want you to write an abstract. Yeah. I specifically say, no AI on this. Versus my seniors, where I'm like, I know you're going to use this to interpret some papers, I know you're going to use this to do certain things, keep it under like no more than 20% of your paper written by AI. And so like there's different levels. It has its place. It depends on what the learning goal is to me.
Yeah. That makes a lot of sense.
Yeah, for sure. Well, because I was just talking to a student the other day, fellow student, and he was telling me that apparently the college is trying to come up with certain AI policies.
Yes.
Protocols.
Yeah.
Do you have an insight into what those would be? Or because I've heard there's certain departments that don't want AI use at all.
In fact, it's because so I have a classmate that's actually part of this project. So they have a lot of faculty that's actually asking students to make presentations on how AI has helped them and how AI hasn't. And right now, apparently, don't quote me on this, but apparently that the faculty split. Some faculties, like what you said, they completely want AI out of the window, and there are faculty that want AI to stay. STEM is the one that wants them to stay. Actually, it's like what she said, that it has its place. If it's helping you interpret more intuitively of a paper, if it's helping you go through difficult parts of a code that's hard to debug. But for the faculty that just doesn't want it to be here is, for example, business.
Yeah, that's what I've heard. But so, yeah, and this meeting was yesterday. So apparently there was 4 students who gave presentations of things that they built, or I believe built, and showed like how they use AI to sort of help build this thing. And I really regret missing it because I tried to go, but I got busy with schoolwork and stuff.
Yeah, I unfortunately missed it. I teach a class Thursday afternoons, but I fall in the middle of that. I'm not anti-AI by any means, because it absolutely has its place. It has its reasons to be there. But I think it also can be overused and abused way too easily. I think at times, particularly your younger, more novice students are going to sit there and go, I can just AI everything and it's fine. And then they get to the point where they can't AI something in an actual job and they have to troubleshoot something, and it's, AI doesn't have an answer for them, and then they can't actually think for themselves. And so that becomes problematic. So I know, and I can speak for the chemistry department, we are very explicit in this assignment, you cannot use AI, this needs to be your own words. This assignment, we're okay if you use it for these things. We have a class that I don't think a lot of departments have. It's called Professionalism in Chemistry, 1 and 2, and it basically teaches you about the field. And we have assignments in there where we're like, okay, you came up with this title, now use AI to give you 3 other options. And so it's kind of showing you what it can do and how to use it appropriately. There's a great AI tool right now, and I wish I could remember the name of it off the top of my head, I can't, but it's for scientific literature, where if you put in a paper and then you say, find papers related to this, and it actually does a good job of thinking about it.
Is it called Research Rabbit?
Research Rabbit, yes.
Okay.
Research Rabbit, because you go down the research rabbit hole.
Yeah. I use that. Very helpful.
I think that's a great example.
I introduced you to it. No, I introduced you to it.
Really?
It looks like...
Oh, yeah.
So we're introducing
Very
that
helpful.
in classes, because then, it's not saying this is what you should think, it's saying these are the topics that are going on. And so it gives you a broader search
and it
engine.
shows you like who cited who,
Yes.
all the different
And how it
references.
goes back.
Yeah, and even shows you the timeline of who cited who.
So I think,
Pretty amazing.
my opinion, for whatever it's worth, as far as education is concerned, both faculty and students, as long as we're clear about our expectations, go for it, right? So like, you can't fault a student for using complete AI if you didn't tell them no AI on this, right? But if you did and you were like, hey, watch it, keep it under 20%, which, by the way, the AI checkers are crud, they can't do anything right. The zero chat GPT and stuff gets things wrong all the time. I will not take an academic dishonesty case based on that. It's not strong enough to do. It's not something I would be willing to put my reputation on the line for, or blame a student for it. So, but as long as you give guidelines, I'm okay with that. And if certain professors are anti-AI, then they just need to say in their syllabus, no assignments should be completed with AI, right?
So I was actually just reading a couple things on X this morning, and there was this paper that was totally constructed, research paper, totally constructed by AI, and then also there was like a new proof for some mathematical thing that was fully generated through AI, and apparently it's correct. And so, have you thought about like what that means for researchers? Like, I mean, it's right now it's probably pretty niche, right? But I've even heard that people are trying to do it with physics and.
Right. It's a tool. I mean, just like any researcher. I mean, conversations that we have with some more mature faculty at times, you know, in their lifetime, they used to teach on overheads. Like, that's what they did. Yeah. And then now it's PowerPoint. And some of them are like, no, anti-PowerPoint because it's death by PowerPoint. I'm like, if it's done right, it's fine.
PowerPoint's going to be beautiful.
Exactly. I mean, you know, you follow the rules, don't go crazy on it, it's fine. No, you don't want to see it with a full page of text. But I think AI is going to be kind of the same thing.
Yeah,
don't go crazy on it.
We used to be like, oh, don't just Google everything. And Google used to be such a big word, right? Oh, you Google that, is probably wrong, right? But nowadays, I feel like it falls to AI. And I think it should be, because AI right now is still very bad with accuracy and all that.
You gotta check it.
Fact check it all the time, yeah.
I know my sister Googled something, which AI is now the first thing that pops up anytime you Google anything. Yeah. And it was something, she was asking about a medication or how to take it or whatever, and she sent me the screenshot and she said, is this right? And I said, what's your end goal? I said, if death is your end goal, yes, that's great. I said, if it's not, I said, I wouldn't suggest taking that like that. So, you know, again, with any computation, I mean, you guys, machine learning, you know this, and it's the same thing we say in research, you know, trash in, trash out. But if it's good data in, then it's going to be good data out.
Which is the problem, because most of these big models out there, they're trained on the entire internet. And let's be honest, there's a lot of trash out there.
Yeah, and there's companies who got in trouble because they were using pirated books and things like that. I just find this conversation really interesting, because I think it really makes us think about
the ethics,
so many things, right? Including like what it even means to be a human, and what the future is going to look like for this, especially when it comes to like academia and learning and.
Okay. Wait, I have a question, because I want to bring it back to drug discovery. Because you know how AlphaFold recently like won Demis Hassabis and a few other people from machine learning algorithms that use AlphaFold to discover drugs, and they won a Nobel Prize in chemistry recently. Physics. Yeah. So, what are your thoughts on AlphaFold 3 and also Alpha Proteo, which is the latest model that they use for predicting completely novel proteins, and then they send it to a lab and they try to synthesize it, and apparently, for example, they've discovered new proteins that can digest plastic faster from like, for example, pollution. So what are your thoughts on like this kind of application for?
I think you hit on exactly the right order that the steps need to be done in. They are discovering and then verifying, because you said it yourself, you send it to the lab, you make it, and you test to see if it actually does what it does. And I mean, computational chemistry has been doing that for decades. I mean, that's the principle of computational chemistry is when you're using it in drug discovery, you will have a 3D representation of a protein or a drug target, be that an enzyme or whatever, and then you will, it's called docking, but you'll just basically throw things at it computationally to see what hits, right? And so then you get like a score of like, you know, this is 90% good, this is 80% good, and then you send that list to the lab, and they either buy the compounds or make the compounds and take the real protein and see if it works.
So my question is, because AlphaFold and Alpha Proteo, they create completely new structures.
Right.
They never tell you how it works, or they don't tell you the dynamics of it.
Right.
They just show you the static end product structure.
I thought AlphaFold was open source.
No, not completely. But because they do 10 predictions a day, they have computational limit. But what I'm asking is, so how does, from your view, because you know how to isolate and what's the other word? Elucidate.
Yeah.
So how would you think about creating these compounds if they show you, oh, this is a completely new compound, never found in nature, okay, it's a completely artificially synthetically made. How would you go about that?
It's worth a shot to make it, right? I mean, you got to try. So you hit on a really interesting point where you said static. And so these models, and as we get better with computational power, these models started out static. They said, here's a crystal structure of a protein. Now, this is what fits in it when it's in that crystal structure. That crystal structure is a snapshot in time. That does not account for the fluid dynamics of the protein moving and rearranging, and what happens if the acidity goes up, or what happens if the basicity goes up. None of that's considered.
None of that is considered in AlphaFold.
Yeah.
All static results.
Right.
That's what people don't realize.
And so you have to be super careful, and that's why you always have to try it in nature, because the amount of times I've seen Schrodinger is the program that usually does a lot of that work. The amount of times I've seen Schrodinger results and say, oh, this is 100% match, this is perfect. Well, yeah, if you were in that unnatural crystal state that they found when they crystallized something, but not in the actual, that it's going to turn off this gene or it's going to produce this
That
thing.
makes sense, because all the data that's fed into AlphaFold is crystallography data.
And crystallography is a snapshot in time.
Yeah.
So the original crystal structure of rhodopsin, which is a protein that has to do with eyes and light and things like that, the only way they crystallized it was when they actually got a ligand, which is a small molecule, to bind to it, and then it was able to be crystallized. But they still have no idea what the actual real structure looks like
That
if you haven't.
That doesn't
have a ligand bind to it.
Yeah, that makes sense.
So you're dealing, and like I said, as we get more computational power, we'll probably be able to deal with that problem.
I don't think so anytime
But it's so
soon.
hard. Yeah, I mean,
It seems intractable.
yeah,
Too many variables.
It's a lot of variables, and I think we finally accepted, and I don't know why it took us so long as a society to accept this, but we finally accepted that every human body is different, so that certain drugs don't work on people the same way, right? And so there's certain people, I'll say it, I'm one of the really weird ones that Benadryl makes me hyper. I will not go to sleep with Benadryl. Yeah, Benadryl's it'll fix an allergic reaction, but it's my last line of defense because I'm super hyper all the time. Wow. So everybody does different stuff.
For me, it's caffeine and nicotine. If caffeine and nicotine, if I take it, I don't feel energetic. I get a headache and I feel like I want to sleep.
It's because you have a mutation in your receptor, same as I do. That, weirdly enough, that's the exact same receptor that works on things like muscle relaxers and stuff. So certain drugs like that do not work on me. And in that case, it's actually genetic. So like my nephew is the same way. But other times it's just a one-off.
Okay, that actually segues into my next question. Okay, it's perfect. Because right now, like what you said, all these studies about drug discovery is mostly in proteomics. How proteins fall, how protein structures are. Now, what are your thoughts? Because right now the new big buzzword, it's been a while, but like new big buzzword in cell biology and medicine is multiomics, right?
Multiomics.
Yeah. So how are you involved in any of that, or do you think that most scientific discoveries nowadays have to involve multiomics? So multiomics just means multimodal data from like different levels for genomics, epigenomics, proteomics, metabolomics. So every layer of reactions you have to take into consideration.
Okay.
I think it depends on. So personally, am I involved? No. I think it's fascinating. I love the whole omics world. Like the omics world is fascinating. But again, it's just data on that single cell line or that single entity that you are looking at, right? So unless we're at the point, which we are getting better about, being able to sequence an entire human, it's not,
They considered a human genome project a failure for a lot of scientists.
Yeah.
They said, yes, we sequence everything, but we have no idea what it does.
Right. And sometimes you're not going to know what it does. Sometimes it doesn't do anything. It's just evolutionary there for fun. Yeah. We still argue about what the appendix is actually for at times. And some, whichever way you want to.
The pinky toe.
Yeah, exactly. There's different things where you're like, oh, it's still there. Who knows why? But as far as how I personally treat omics, I treat it like a large data set. Like an omics project at all of those different levels, to feed it into machine learning would be amazing, because then it can see it can extrapolate more and see what kind of hypothesis or theories that it can come up with, but then we still have to test it in the real world. But
grounded, right?
But I love the omics and everything that we do. I just think the more we can analyze it, that multi-omics, bringing in AI to analyze and help us see the connections that we miss, I think is great.
Because the holy grail of bioinformatics right now is to put all the different omics together and hopefully find a bigger picture.
Yes.
Because they say that if AlphaFold already found so much, imagine if we put other omics together into the data.
Yes.
But then again, it's like what you said, most of the data is a snapshot.
Yeah.
We don't.
So is the dream in sort of pharmaceuticals to like
Personalized medicine.
Personalized. So how would they go about that? Would they take your blood and then be able to put that thing in a petri dish and then inject different drugs into that thing and see how it reacts? Like, because I've heard this idea of essentially like a, I don't know any other way to say it, but like a body in a petri dish, like
Organoids.
I think, and you're starting to see the shift in certain fields of medicine now, and I'm happy that we're starting to see that. When even 20 years ago, 30 years ago, it was we treated cancer by where it was in the body. You had lung cancer, you had brain cancer, you had ovarian cancer. That's not how cancers work. Cancers work better when you know what the mutation is that caused the cancer. So you could be a RAS mutation in the lung, but you can also be a RAS mutation in an ovary, and you should treat those with drugs that treat RAS mutations, and you would have much better success rates to treatment. So now it's almost standard practice when you find tumor cells or cancerous cells that you biopsy and you sequence, and then that's going to tell you what medicine's going to do best for you. Another part of that, and why it hasn't gotten into, like the common cold or the common virus, is because people want results right then. There's a very interesting study, it's several years old now, but patients do not feel that they spent a good amount of money or were valued if they do not walk out of a doctor's office with a prescription. They don't think it was worth their time, which, that says a lot about drug culture in the US and everything else, which we're still better than just handing out antibiotics in Europe, which is what, you can buy penicillin off the shelf in Europe.
Oh, wow.
That does not sound responsible at all.
That's not,
No, especially
Europe, drug resistant. Europe's had issues with that for a while. But, so, that's always been so funny. I'll have international students that I've worked with along the way, or when I was in grad school or stuff, and they're like, oh yeah, I'm just going to go pick something up. I'm like, what are you going to pick up? And they're like, oh, just some azithromycin. I was like, you ain't getting that without a prescription.
What is that?
Z-Pak, it's an antibiotic.
Oh, okay.
Yeah, because they're like, oh, I think I have bronchitis or something. And I was like, you can't do that.
Yeah.
We take antibiotic stewardship a little bit more seriously than that. But yeah, you're not going to get to the point where we would sequence, the susceptibility of every, urinary tract infection or every lung infection, because people want results right then. So you treat broad spectrum to do what you want. Although if you knew exactly what it was, it would be way easier to treat it, and it would be a shorter course to treat it. So you still have the human dimension, for good or bad.
I've talked to a few people, even at my workplace or just random strangers that bump into in the street. It's I usually have this short coffee talk with them and ask them about their views on a lot of things. A lot of them seem to, either through conspiracy theories or either through just their experience with healthcare, is that they call it, most healthcare nowadays is sick care. They're treating the symptoms, not the causes. And then I give them the idea of preventative medicine, right? They say, what if it's your lifestyle? What if it's just, like you said, genetics? You just need to treat that instead. And then I ask them, now, how do you quantify that? You prevented a disease, but no one celebrates a war that doesn't happen.
Yeah.
People celebrate a war that's ended or a sickness that has gone away. No one celebrates the fact that, this is my third year that I haven't gotten the flu. No one celebrates that. Yeah. So how do you quantify a non-event that hasn't already happened?
And that's the hard part. How do you convince people
to fund preventative medicine?
And I'll give you an example without sharing too much data. My mother is the youngest of 3 siblings. She grew up in rural Virginia, Western Virginia, almost West Virginia, in the Appalachian Mountains. So not a lot of great healthcare. There was one old doc that was there. Mom's also 71 right now, so this is a while ago. And she lost her older brother and her sister much younger than 70, where she is now. They were actually younger than what she is at this point. And it was because they stayed in the area. They had a little bit of a harder life. They both succumbed to strokes, coronary artery events, things like that. As where Mom, when she was in her 20s, stepped out, joined the Air Force, met my dad. Dad was in the military for 24 years. Mom got out when she had my sister to be a stay-at-home mom. But the military is big on preventative care. They are big on making sure, if your blood pressure's up, let's go ahead and medicate. If your cholesterol is high, back then it was over 200, now we use a different metric. But they're like, let's get that down. And so Mom, knock on wood, has never had a coronary event, a stroke, a heart attack, anything. And she lost both of her siblings to that much younger. So preventative care works. I mean, it is what gets you there. It's just socioeconomic issues play such a role in preventative.
And also like incentives, and also how do you how do you perceive and quantify a non-event?
And is the reasoning behind the military doing that to keep your troops healthy? Like what?
I think it always was, and just ready access to healthcare. So when you're in the military, you and your family have free healthcare. So like it's not I've got to make a copay or whatever. It was if you were in there every year for a physical, or you were in here for whatever happened. So I think it's just the difference in different types of medical care. Same genetics, but...
What are your thoughts that medicine, healthcare, and also, education should be a universal right? It's just like a thought that popped up because I was talking to a guy the other day, and he's like, one of the major problems is the system, right? He says, and then he says that it's the incentives, capitalism. He says that education and healthcare should be a universal right. So what are your thoughts on that?
One way above my pay grade. I'll say that. I will also say there is no doubt that education levels are tied to healthcare, preventative healthcare. Doing better economically means you do better in a health-wise. It's not a surprise, I think, to anyone that when you are a state that is lower in education, you're also a state that's lower in health, right, in general.
That's true.
Those go hand in hand statistically. There's a saying about causation is not
correlation
is not causation, but in that one I think we can make that jump probably. Most of the time I warn against that. So I don't know, I go back and forth on that, and I'll tell you why about a universal. So the advancements that we make in the US are probably the best worldwide. We have the best, outcomes. We have the best medications. We've got the best clinical trials. There's a lot of innovation that is happening in our medical system. And
that is because of capitalism.
Capital incentives. So for the good or the bad, would we have that if we had universal? I don't know, because we don't want to lose the innovations and the new cures and the new everything that's driving the system. But the fact that it's just broke my heart to see how much the new sickle cell treatments were. I mean, you could cure sickle cell with, I think it's like 2 doses of this, but it's like $6 million a dose.
What? And also the diabetic treatments too, right? They say that around the world you can probably get it for like less than $5, and here you can get it like...
I've heard it's like pennies on the dollar to actually make.
In Malaysia we get it for free.
Wow.
Almost for free,
Now, I didn't listen super close, but I did feel like I heard something on one of the news shows the other day about trying to lower medication costs, but the federal government was doing that, so I don't know where we stood on that. I was on the drive home yesterday, and I was slipping through channels, and I happened to stop on that, so I don't know where that's sitting right now. But yeah, I think the statistic I heard that really shocked me was the U.S. has 4% of the world population. We use 13% of the drugs that are produced in the entire world, and we pay 70% of the pharmaceutical industry.
Wow.
So those numbers, I haven't fact-checked those numbers. That was something, I'll clearly say I have not fact-checked those. I meant to go do that today because that's very interesting.
Been known for a while, though, that, there's just, here in America, it seems like we just look at it as there's a lot of money to be made because people need these things, right? And I think, other countries, governments try to do, I guess, they just try to go a different route whenever it comes to pharmaceuticals and getting people the medicine they need. So.
Well, I remember working in grad school on, it was that malaria drug project, and we were working on the synthesis of it, or partial synthesis of it, and we were talking with some agencies, and they're like, you've got to get it to where we can make it for, it was some pins, like $1 a pill. Like if you are not under $1 a pill, we are not getting it to the countries that need the malaria treatment, because they don't make $1 in an entire week, a US dollar. So like you can't, you have to get it to the point where you're going to be okay.
But then again, even if you make it $1, they probably won't sell it for $1.
Right. Well, and it was a project, I think, that we were looking at funding from the WHO, the World Health Organization.
Okay, that's fine. World Health is...
Yeah, and World Health was like, we've got to get it to where it's under $1 to be able to even consider being able to make this.
So, one thing that I've gotten into lately is these things called ketones, exogenous ketones.
Okay.
And so apparently, like the military spent billions of dollars to try to come up with this way of giving sort of a energy boost to their troops, and so they came up with these exogenous ketones. And ketones, apparently what our body uses as energy. But these things are still pretty expensive. It's like $4 or $5 for just a little shot. I forget exactly how many ketones are in these things, but it's sort of interesting the energy it gives you. And so I was wondering, like, if you sort of knew maybe why they're so expensive to chemically manufacture.
I don't know. I haven't heard of this before, so I'm gonna have to go back and look at that. But it probably depends on what ketone and the purity that they're looking for. So some of those reactions, particularly like, you wouldn't think it's cost prohibitive to make a carbohydrate, which is just sugar, but to get a pure carbohydrate that is a single carbohydrate is horribly expensive. The purification process takes forever. So I would imagine it's probably on the purification end of it, of what they're making. But and this happens with a lot of drugs. I don't think people realize it. You have a molecule, and I was kind of talking about like that 3D structure, like there's a stereochemistry with an R and an S version of it. A lot of times in pharmaceuticals, like one version will be active and the other version will do nothing, but you don't bother actually separating it out because it's not cost effective to separate it. Classic example is Xyzal and Claritin. They are the exact same molecule. One is a racemic mixture, means it has both compounds in it. One of them just doesn't do anything, and the other one is a pure mixture.
Okay. Is Xyzal not xylitol or xylitol, which is a gum, right? Or something like that.
Different compound. Yeah, different thing altogether. And I was using trade names, but because I can't think of the actual compound name on top of my head. But it makes a difference there, right? And so I would assume it's kind of with the purification issues.
Interesting. Yeah. So I would like to get back to this topic of sort of building a research lab.
Yes. Okay.
Because it's kind of something that I'm you know,
we're nerds.
it may be
want
starting
a
to
lab.
dream about.
Yeah.
Right?
We all want our own lab.
yeah. So, you know, maybe what are some things that you ran into that you that were maybe unexpected, and like maybe what were the you know, the hard parts to trying to build this lab up?
So I will say some struggles that I've seen other people have is finding students. I was very lucky in that my research is appealing to you guys. Students love my research, and so they like the idea of the pharmacy and the natural part of things. So I have drug discovery. Yeah. I've never had a problem finding students.
As soon as I believe it was you that told me what your research was with, I was like, okay, that's interesting.
No, I have to say, before this talk, I've never liked chemistry more.
Yeah.
God damn, that you're able to make chemistry sound so interesting.
Yeah, I mean, one of the questions, you know, I wanted to ask, but you sort of just explained is, I wanted to ask, like, well, what to you is the importance of chemistry? But
can you
throughout this whole thing,
everywhere.
you've sort of said that in so many words.
Because
right?
initially we were joking about how, like, okay, out of all the physical sciences, natural sciences, we're like, okay, what's your least thought about one? And we're probably like chemistry. And then we're like, one of the questions we should ask Dr. Waters is like, how do you make? Can you convince us that chemistry is important? But you didn't need to.
No,
you already did.
It's there.
You already did.
It's there. And again, interdisciplinary, right? It's how it relates to everything. Often when I'm teaching my very first lecture of Gen Chem 1, and I was like, okay, chemistry is fundamental. You can't do biology without chemistry, but you can't do chemistry without physics, and you can't do physics without math. So we all go together.
They're all natural philosophy.
Yes, all natural philosophy. So sometimes that's a struggle for new instructors of trying to find the interest there. Sometimes I'm kind of broad in what I do. I'm also kind of very open about my research. So I have the Marine Sponge Project. That's what a lot of my students do. That's kind of the fallback. But I'll also have students come to me, and I had one 2 semesters ago. Great, amazing upper division student, and was like, I want to see if there's any anti-cancer compounds in lion's mane mushroom. And I was like, go for it, let's do it. And so we bought powdered lion's mane mushroom, we bought dried lion's mane mushroom, she even grew some fresh lion's mane mushroom, and we were testing all the different ones and seeing, and the interesting results we found, you know, we're close to isolating the compound from that.
Just
that we like.
From the fruiting body? Or
From the fruiting
you've had to go through like an extraction, right?
Yeah, you do have to go through an extraction, yeah.
Because from what I've seen is like, so I take lion's mane as a nootropic, right? But from what I've seen is like, you don't want to take the fruiting body. That doesn't have the actual nootropic in it. You want to do a hot water extraction and a ethanol extraction, and then combine these together.
We actually did, because generally up until this whole GLP-1 revolution that has happened, you generally, when you were doing pharmaceuticals, you only wanted small organic compounds, and that generally leads you to an ethanol extract. So it used to be, unless you just were really a glutton for punishment, you ignored the water extract to a certain extent, because it's going to have a lot of sugars, peptides, other things in it. Not saying you wouldn't go after those, but it's definitely not easy to go after those. And so we generally did the ethanol part of ours. Or alcohol mixture.
Interesting. Okay, so what are some other things that you've ran into?
So I think the question when you first start a lab, and it's different whether you're at a research intensive institution like an R1, or if you're at a PUI like we're at, leaning towards R2 by 32.
Yes. I want to talk more about that later for sure.
Or if you are, more in a community college. So to me, this is the perfect balance of, I can do some research, but I'm also teaching, and so I can make those two happen together. So I love this balance. I said in the beginning, I teach usually 3 to 4 classes a semester, so I'm still very active with the students. I know the students, I still run a research lab. Finding that sweet spot of how many students you can have without losing your mind is a very interesting number. Over the last 10 years, I've kind of found where that number is. It's right around 5. 10 is too many. Found that out. That was also the semester I was in
Oh my gosh,
COVID.
yeah, I can imagine.
Yeah, that was because you're trying to keep everybody's project straight and you're losing your mind slightly.
Yeah.
But you live and learn.
Yeah.
Because you don't want to say no, but sometimes you gotta say no. But figuring out what is there and what you're going to need and what your startup fund is. So generally when you accept a job with some type of research component to it, you have to, they'll give you some type of startup fund. It depends on obviously the college, it depends on the department, it depends on
Your grants they have.
Your grants you have, kind of where you're at the time. And so UCO is very good about collaborative research within the departments, but also outside of the departments. So in more research-intensive institutions, you get an empty lab and say, fill it with what you need. So that could be you have to buy all of your instrumentation, your mass spec, your
NMR.
Well, usually there's a core facility for NMR because that's a little bit much, but $250,000, that's a little more than people are willing to go. But at UCO, I was able to call and say, okay, what do you guys have? And then I was able to put together what I would need to start with and go from there. And then there's always grants that you can write. We're very fortunate to be an NBRE state. So NBRE is funded by NIH. And so it's basically for states that do not have enough biomedical research that could do more, that could develop more. And we've been an NBRE state for a few decades now. And it's very nice because it gives the award from NIH goes to OUHSC, Oklahoma, OU Health Science Center, but then they have specific funding that comes back to the 4 years, not including OU and OSU. So we're not competing. So we compete as regionals, and UCO takes the largest chunk of the regional because we have the largest amount of research of the regionals right now. Not that the other regionals. Well, we're the big. The whole university system is an interesting concept of where
the border regions and all that.
Yeah, I mean, we're not as big as OU and OSU, but the next biggest regional is only like 6,000, and we're at 12, 13,000 students, so we are just in a weird category. Always have been, and I prefer us to be a big fish in a small pond as opposed to a small fish in a big pond, but that's kind of where we're at the moment. So anyway, good funding. So I've personally benefited from NBRE equipment grants for a lot of stuff in my lab.
Yeah, because for example, one of my professors, he says that he's been to OU and OSU, and he's been to here, and he says out of all three, he loves it here because it's like what you said. He said he's able to balance teaching and research, and it's not too intense on research. The grants are not too high expectation, and of course the NBRE part that comes into play. And I'm just curious because sorry about that.
No, you're good.
Because I'm a biomedical engineering student, right? And I have a lot of people who ask me, like, okay, how can I contribute to science? Because there's so many ways. How do I know what I want? But I think a lot of students don't understand there's more than one ways to it. Contribute. You can either discover a method, verify a better method, you can do that data analytics, or you can work on a new model, a new theory. So can you just briefly go over that? Because I notice a lot of students don't know what does it mean to contribute to science. They think that you have to win a Nobel Prize to
It goes back to intrinsic motivation. So what is going to keep you driving forward there? So at least for me, like I talked briefly about kind of not giving up the bench, but giving up as much research as I was doing to take a position here, and never looking back because I love it and I love the balance. And I would never want, personally, it's not for me to be at an Art 1 institution. It's not an environment that I really like being in. And I can say that as someone who graduated from OU and worked in the research labs 4 years there. Weirdly, none of Robert's graduates went R1. Several of us went PUIs, but none of us went R1s. He's starting to get a complex about that. We said, well, you made it look so appealing. But there's several of it, like 5 or 6 of us now that are PUIs, but none of us have taken them up on the R1 thing.
What does the PY stand for?
Predominantly undergraduate institutes. Yeah, so very few graduate programs, and usually not any PhD programs, although we're
in between. That's what R2 by 32, right?
Yeah, we're in between on that, which
and I know they're
is
working on more PhD programs.
Yes. So
you seem to have two sides to that opinion.
It's a complicated topic. It's a really complicated topic. It's doing what's best for the student population that you have,
and also the institution,
and
right?
Institution, yeah. And so you're balancing a lot of things there, but yeah, like I said, intrinsic motivation of what is a contribution to you. Some people are going to be, you familiar with the show The Big Bang Theory? Yeah. Some people are going to be Sheldon, and they're going to think that's if they don't get that, then it's not going to happen, right?
They're going to die.
Yeah. So that's what you're like, sweeten or die, and that's kind of where you're at. But it's one of those where no contribution is too small, right? Whether that is, a science ed major that's going to go out and make those contributions to the next generation of students.
Like a scientific communicator.
Like a scientific communicator, like a K through 12 teacher. Kudos to anybody who can do chemistry to K through 12, because that terrifies me. And I say that as someone who has a 17-year-old nephew in chemistry right now. And he came home and said something, and I said, don't do that to your teacher. I said, please no.
No.
I was like, no, we're not playing this game. So I think the words "don't make me come sit in that class" came into play. But so he's wonderful. I'm kidding. But there's a lot of different ways you can contribute. It doesn't have to be, the biggest and the best. Some people are geared that way, that okay, this is what you're gonna do. You're gonna be, the person bringing in $6 million, that's a light estimate, $20 million worth of funding from the NIH and NSF every year. Like, some people are geared that way. Some people are geared more towards, it's important to me to go out and do science Saturdays at the Science Museum and get, the love of science at a young age, whether that's chemistry or physics or anything. So it's all contributions are valid, and I try to make sure everybody understands that. I've heard the question phrased to me before, not necessarily by students, but by colleagues or others, and they're like, well, why'd you settle? I was like, I didn't settle. I love what I'm doing. What are you talking about, settling? I've got the best of both worlds. I've got students who are going on to make a huge difference in the world and contribute everywhere, and then I've got me still doing some research here and training the next thing. I was like, I didn't settle. So I get annoyed with that question at times. But
I mean, UCO is great.
It is. I love it. I wish I would have known it existed when I was applying for colleges. I don't know if we just didn't advertise in the southwest part of the state, but it was, you heard OU, OSU, or Western, which is a community college.
I did not regret coming here.
Yeah.
So R2 by 32?
Yeah, exactly.
I look at his face. His eyes tell me about R2 by 32.
Okay. So let's try to
unpack this real quick.
Okay.
So how do I want to start this? So I'll tell you maybe what my thought is, and then maybe you can tell me whether we should sort of slow it down a little bit, right? Because and maybe this is just me sort of projecting a little bit, right? Because I've always been a big dreamer, right?
Yes.
I come up with these grandiose ideas, and I just go forth and do, right? So, research has done wonders for my life, right? It's opened so many doors. It's opened new paths in my brain, right? It's made me find the thing that I truly love in life, right? If I can keep doing research for the rest of my life, like, I'm totally down for that. And
That's how I felt.
And you know, there's professors like you, and Dr. Benjaber, and Dr. Teo, and Dr. Lamachani, you know, that sort of give me more fuel to go in that path, right? Because they just make it look so fun and interesting. And so I want to give back in some way to UC. I feel like you know, because I may be going to OU if I get accepted, right? But then I also have the option of staying here, right? But so this whole research thing has just wanted me to just make research more available to students, right? I just have this dream of just students like all the students knowing about the research going on around campus. And because, you know, we come across students all the time who are interested in research, but they don't really know how to get involved. And honestly, if they don't come across me or Albert,
they don't know
I sort of worry if they ever come across the right person to talk to.
Yeah.
And so my thought is, well, there needs to be an easy way for students to come across research. Mm-hmm. And my thought is, well, you know, our website should be, you know, research should be easily accessible there. Where, you know, right now you have to sort of like go searching, right? So what do you think about there being like a header in the front homepage and it says research, right? Because I know you said you have reluctance for like R1, or just it's a different environment, right? It is. And it's not for everybody, and it's probably not the type of environment I want either, right? But and so how do I want to say this? So is that like the vision for R2 by 32? Yeah. Right? Is that in your mind? Is that getting us sort of further away from something that we have right now, or is that actually good for our future, or?
Not so. I absolutely support R2 by 32. I support that, but like, as engineers, you know, if you're going to build something, you got to have a good foundation. And so there's a lot of questions I think faculty have at the moment of what does more research look like with the teaching loads we currently have? What does more research look like with the funding that we currently have? Everybody wants more research. Absolutely. All for it. But we don't want to grow too fast and lose the connection we have with the students, right? And so it's a balancing act that I think we can do. I think I am very glad that President Lamb is as enthusiastic about it as he is, and I'm glad that he has a vision and a mission for where we're going. I would like to see a little bit more detail of how we get there and still maintain who we are, as far as that's concerned, because I don't want to lose the connection with you guys. I don't want to be teaching a 300-person class, right? That's not what I want, but that's occasionally what happens when I don't have enough teaching capacity and research capacity at the same time. So one of the main differences between an R1 and an R2 or even a PUI like we are is that class sizes are small. Even my labs, for the most part, all my major chemistry labs are taught by professors. They are not taught by graduate students. That's not the experience I had when I went through OU. I had, a professor, Robert might have taught one class a year, not a semester, a year. Wow. And so, I had 300 people in my Org 2 class. Mark Morvant, who I know well now, couldn't have picked me out of a crowd, right? It wasn't a thing. So, I had physics, again, in a class of 300. So 1 and 2, which was not super fun. So I don't want us to get to that point where we lose touch with the students. I'm all for more research. I just need us to see how we get there safely and grow at a manageable pace where we don't lose who we are.
So, because I know that like the homepage of a university sort of tells you exactly what the is supposed to, right?
Yes.
It's supposed to sort of show like what the university is about.
Yes.
So is putting research on that front page, is that?
I think that's great.
Okay. Like,
I would love to see a link on that front page to the RCSAs that our Office of Research and Sponsored Programs do, so that they can go straight to the RCSAs. I'd like to see, now that you got me thinking about this, I mean, like, I'm very familiar with the RCSA page. Also, shameless plug, RCSAs are due February 20th, if you're listening to this. So...
Is that for graduate students too, or is that just
graduate students can apply for RCSAs. Yeah, absolutely. So I would love to maybe even see a link to some of the research centers, like maybe back to CIBER or back to CFACS or back to CREIC on the RCSA webpage itself. Also, I think it's really important not to forget that CMS is not the only ones doing research. There's amazing research going on in the psychology department, in the anthropology, sociology, education, business. So they don't submit nearly the number of RCSAs that we do, or us in FSI. But I think it's important to understand that there's that research as well. So how can we make sure students, and I don't know, I'm throwing this question out here, how do we make sure students understand research isn't just a beaker in your hand, right? How do we get them to see no matter what college you're in, what area you're in, you can be involved? Like, that's the important part, I think.
That's, I think, a big mission of ours, right? Is trying to tell other students about it. We talk a lot about our research in this podcast. We, talk about, just like research may sound scary, but it's not scary at all. Like, you're literally just learning and then trying to make a little incremental progress, right? And especially as an undergrad, you're not having to come up with your own idea or anything like that. Typically, the advisor has something in mind, and so that's why I think it would be interesting to have like a big link on the front page where you could just press research, and then so someone like MIT, right? I've gone to their website and they have this, right? And then the next page it takes you to is all of the departments that do research. You can click on the department, that takes you to the department's page. It tells you their mission, and then you can go to, I think there's a link for all of the people involved in that, and it goes through the board members and everybody, and you can eventually get to one of the professors who's doing research. And so you can go through and like, I've always had this interest in psychology. Let's see what kind of research they're doing. Go and find, the professor and look at the papers that they've written, what his specialty is in, and maybe you find that, man, that sounds really interesting. So let me email him, right? Because sometimes email can be the easiest thing, right? One of the things that we try to do here, and what we're trying to do today is show that our professors are humans too.
Oh, absolutely, absolutely.
And so we want to make it. We want to make. Hey, what's up, Iris and Wes? We just want to, unveil the mystery, because sometimes professors, we can look up to them and we can sort of be a little scared of approaching them, right? And so one of our missions is to unveil that and show that they're human too, and they're just as curious as you are. So go and like have a conversation with them, but sometimes an email is easier though, right?
Exactly. And that's so important, and I tell my faculty this all the time. I was like, tell them what your research is in your classes. I was like, let them know what you're doing. I start every semester when I talk about me and like where my journey is and where I've been. I was like, and here's the research I do, and if you're interested, come talk to me. Even if you're not interested in my research, come, I'll find somebody else you can research with, right? And making these connections. So it's, the biggest piece of advice, if you want to do research, just go talk to people. It might not be that first professor, but they might put you in line with the next one or the next one. It's so important. But I love that idea, and I'm going to give you a contact when this is over and see if he can help you out with that. Yes.
Yeah, because
Also, he might be a great guest on here too. So.
Okay, awesome. Yeah,
That'd be awesome.
Heck yeah. Yeah. You know, I just want to make it really easy, and I think that would be the way. You know, even let's say somebody's not even interested in research, but they have to go to the homepage of UCO to find something else, and they just stumble upon that link, and they're like, let's click on that. And then also, I would think it'd be really good for people who are trying to give funding to UCO or thinking about giving funding to UCO.
Absolutely.
People who give out grants, right? They could go and see, okay, well, how serious are these people about doing research?
Yeah, and most research papers, they show their grant funds.
Yes,
So-
you have to. Yeah, absolutely.
Yeah, and you know, really, to me, I mean, you know, I'm no software engineer, but it just doesn't seem like it would be that. The pages are already there, right? Like your page has your bio and your information on there, your papers and everything, and so. I would just think it'd be easy to just link those things together. I mean, maybe not, but
I would think it would, and that's why anytime I get a chance to go out and we're talking about cyber, we're putting up tables and things. I have my cyber faculty. I was like, look, if you're looking for researchers, give me your one slide, and I lay all the slides out on the table. And I was like, any of this research look interesting? Here's the email, contact them, and trying to get that out there. So I always tell any of my faculty, I was like, there's no reason not to have research students because they want it. They're hungry for it. They want to be there and they want to be involved. So.
yeah, and you know, now there's CURB, which is to help highlight undergraduate research.
Yes.
And so, you know, maybe on everybody in every professor's page who's doing research, right? You link it to CURB or maybe the professor or the students that are under them, right?
Yeah.
So that you can go and look at what are the students doing that are underneath this professor. So,
Look at that. Yeah.
so this. So why does what is really the big difference between PUI and R2?
So
Is there like a structural difference? Is like a
not really, to
are we
be
gonna
honest.
have to pay more to be here, or like what?
So all of this, and it gets complicated at times because all of this goes back to Carnegie designations. So you hear the word Carnegie. Carnegie is the way we set up all of our American university systems, and that's why we do credit hours the way we do, and this is why certain classes count the way they count. This is all part of the Carnegie system. So Carnegie has classifications, and one of them is an R1, one of them is an R2, and I think PUIs kind of fall under R2s at times, but there is a certain demographics that you meet. There's a certain amount of money that you are bringing in, and I'll have to double check myself because there are people on campus that are much more informed than I am about this, but I think we already pretty much bring in that amount of money.
I see.
It's just a matter of having the right number of graduate students and things. So I think in talking to almost all faculty, it's not that anyone sees it as a bad thing. We want it. We want more research for the students. We want that. We just don't want to fundamentally change what we already have as well, right? So we're all great with growing as long as we grow with care. And so I think we all support what's going on there and how we do it, just making sure we don't lose our identity in the process.
I think the keyword would be manageable growth.
Manageable growth, exactly.
Because if it grows too fast, you guys, like what you said, you'll lose touch with the students,
Yeah.
which is what a lot of professors here love. You might lose a lot of time either for research or for education one way or the other.
Right.
Because we might become too intensive for research. That makes sense, honestly.
So is R2 by 32? Like, for you, like, do you think that's enough time for us to be able to iron out and make sure that we don't lose ourselves? Because what is
think
that?
if
That's
it's
six years from now?
I think if we're done carefully, I think we can do it. Now, I say that as a department that doesn't have a grad program. So chemistry actually does not have any graduates, grad programs. We don't have any masters right now.
Do you want to have?
It has to be in the right thing. And you have to do what's right for your area. So there is exploration. Most people don't know this, but there's a very large biotech core that exists within Oklahoma City. There's Cytovance and ARL and many, many other biotech corporations. So an analytical master's might be our ticket. But it has to be a specialized master's. It can't be a general master's.
Yes.
So it has to be the right thing.
I'm just curious because, for example, I heard from the idea that the Board of Regents would also care that, for example, OU is well known for this, so they will fund more for that. Or OSU is well known for this, they fund them. They don't want to let two colleges compete with each other.
Yes. So what we have to choose is
our niche.
Yes.
So aside from that analytical one that you talked about, do you think there's any other things in chemistry, for example, or in CIBER as a program?
Right. Well, and we have discussed that. There has been discussions in the past of it's rare. In fact, I only know of one on campus, but the Professional Masters of Science, PSM, Professional Science Masters, actually resides under CREIC. And it's the only one to reside under a center and not under an actual department or college. So it like jointly resides under a couple things. So there's been talk of maybe interdisciplinary masters coming out of it. We're seeing, because maybe even like lab management is a big thing that people need, and so maybe they want a little bit more education than their bachelor's, but they don't want to go full-on doctorate. So it's, where do we serve the Oklahoma City metro the best? Because we're a metropolitan university. Where does that come from, and where does that look like? And there's a lot of discussions that happen at the faculty, college, department levels all the time of where is that happening.
So I guess maybe I'm confused on what's the difference between a master's and a PhD.
So at a couple of things. One thing that also is important to note, and I've mentioned the term PUI and we've talked about it a little bit, there's special grant funding for PUIs where we don't compete with the R1 institutions. And all of us are, you really want to make sure we don't lose that designation by having too many graduate programs at a time. So that's, I don't think we'll get there. I think we'll be totally fine being an R2 with that, but that is something that is always kind of in everybody's mind of, again, talking about big fish in a little pond, little fish in a big pond. If we lose our PUI designation, does that shoot us in the foot and make us have to compete with the big boys, which is harder because we don't have the resources that they have at times. So
manageable
scaling, right? And that's the important part. So the real difference between a master's and a PhD depends on your field, to be honest. I mean, yes, there's an educational difference, like I talked about, 5,000, 6,000 level classes,
and also
thesis, dissertations, those types of things. However, certain fields, one is more prevalent than the other. In chemistry, to be honest, not a lot of us have masters. We almost all have PhDs. So it's just how we focus.
So, just a quick segue here, because you said an analytical chemistry master's is good for UCO. What are your thoughts on a PhD program of that in UCO?
By 32, it's going to be a stretch for our department because we don't have a master's. I think it would be a stretch for us. I think so. Now, engineering that already has a master's, biology that already has a master's, that's a bigger potential to be able to do it, depending on what the master's is. The PhD is. But for chemistry to jump straight to a PhD by 32, I think would be difficult at best.
Is it because of curriculum, or because you said most chemists goes to PhD anyways?
Right.
So
curriculum-wise, it's also a thing as well. So right now, we have dualistic classes, 4,000, 5,000 level classes, but no 6,000 on the books. And you cannot dual teach a 6,000 and a 4,000 level. So a 6,000 has to be its own thing, which means my teaching capacity has to go up. And so I have to have people to cover that class. Yeah. And so again, it's part of managed growth. Yes, we want to get there, but
so
we gotta be able to teach the classes we need, right? So it's all kind of falls in, and it's a very complex problem. I think
it's a very logistic problem.
It's very logistics heavy. So like I loved how you explained it the other day, just a little bit ago, where you were like, okay, I want the big idea, I want this new thing, I want to see it, but you also got to implement it, right? It's great to be the idea person, but you got to implement it well as well. So, like I said, fully support it. I think it's I want more research. I want more research for our students, but we've got to get there in an appropriate way, which I think we can in some aspects. Now, it's interesting, very old history here. The master's program that chemistry had, and this predates me by quite a while, was forensic science. Forensic science, the institute grew out of chemistry and became its own institute, and when they left, they took the master's with them. And so that's why we used to have a master's in forensic science, and now it's after that split, it was kind of, do we create another one? Where is our niche? So we had a niche at one point. And then it went kind of away.
Why not just make it a multidisciplinary, like what you said, like interdisciplinary PhD? Or
I believe that is what they're trying. I think I've seen some paperwork to see whether that's going to be, and again, it's almost splitting hairs here, whether that's going to be a PhD or a Doctor of Science. So right now we don't have PhDs on campus. We have a Doctor of Science.
Now what's the difference in that? Now, that seems to be more of a gray area.
That is much more of a gray area of what it is. So technically, and I'd have to double check, and so please, if you're looking it up, double check me here. But I would say that the Doctor of Science is more practice-based, as where PhD is more Doctor of Philosophy, research-based-ish. At a certain point, though, they both get you the same job, so it's not super different. But we technically have Doctors of Science. We don't have PhDs. I think there might be applications for us to potentially do PhDs in the works right now. But again, that's just changing some designations. And then one thing that we kind of haven't talked about, but I think would be great for the university, is if our nursing program can have its Doctor of Nursing. I mean, a nursing practitioner doctorate is an amazing thing too. And that's another area of growth for us.
Oh, interesting. So Doctor of Philosophy, PhD, is just a wide range of disciplines, including humanities, engineering, and arts, not just sciences, but they all focus on originating research and knowledge contribution and distribution, often academic oriented. But Doctors in Science is just primarily STEM. That's practically it. But in US,
they're not as common in the US.
No, in fact, it's more common in Europe and UK. Interesting. So what made us decide to put that? Is my question.
I was not involved in those conversations.
Do we have European origins?
Well,
again,
are you talking about a country?
Again, it kind of, I think, goes back to, you hit on it where you're like, well, sometimes OU has its thing and OSU has its thing, so
we want to make our own thing.
It's kind of our own thing, and they weren't probably going to argue with the PhD. They weren't going to argue with the Doctor of Science, but they would have argued with the PhD. So one thing that I don't think people realize as a whole, I didn't until I started being involved in this process, is if you're going to start a new degree, or let alone a new level of degree, you have to have approval from RUSO, and RUSO is our regional university system, so it controls all the regionals. So everybody likes to say the compasses, Northwestern, Northeastern, Southwestern, Southeastern. And then you also have to have approval from OSHRA. OSHRA is everybody's region in the entire state. And in order to get their approval, it has to go out to the other universities and colleges and see if anybody objects. And depending on the level of their objections, so like at times where you're talking about not wanting to compete, like let's say, UCO wanted to start a pharmacy school, okay? We would get a lot of pushback from Southwestern and OU because they both have pharmacy schools, and they go, do we really need a third in the state?
Yeah.
So there's a little more politics in it than people realize.
Dang. So when this thing comes up about R2 by 32, were you like notified about this before, or was this just like a or did
Fall
they talk?
forum? I think we heard it at Fall Forum.
Okay.
I want to say that's where we heard it first.
Okay, and so how does this come? How do you think this comes about? Like, is it because from what I've heard, is that UCO is a large school,
Yes.
but we don't get funded in proportionality to our size.
Correct.
And so is this what we're just like reaching is the wrong word, but are we just trying to get some more funding? Is that...
We are a state-supported school, not a state-funded school. If you look at the percentage of our budget that comes from the state, it is not enough to actually
What does that even mean? Supported and not state funded.
The last numbers were like 13% of our budget is from the state. It's not very much. So
most of funding still comes from IMBRI and all that stuff.
Or is it
tuition.
private?
Tuition.
Oh, tuition.
So we're mostly self-funded.
Yeah.
Oh, interesting.
And I'm surprised that our tuition's relatively low compared to
It is.
other. Yeah.
It is, and that's a big thing to keep it that way.
No, yeah, but now that goes back to the question: if the state doesn't want to fund us, but we're self-funded, and our self-funding is slower than other people's self-funding, which is already higher than most.
So
The finance,
confusing.
the
Okay.
accounting.
So here's how it actually happens. There is a state appropriation that comes down from the state legislature and says, Higher Ed of Oklahoma, OSHRA, who controls everybody, you get this amount of money. Okay? Oftentimes, in addition to that, there are certain factions that will lobby for an additional appropriations bill for OU independently and OSU independently, and they will get other funding, and on top of what OSHRA would give them.
So do we do that?
I think we're starting to try. We saw $1 million go to the engineering department. We saw nursing get a fairly large chunk. We saw teachers prep, which is the education college, get a fairly large influx of money. So I think we're getting in that direction where we're asking for it, and they're noticing that UCO is the bigger one.
Let's go.
The way that Russo goes is, so the money comes down from OSHRA, splits out to the OSU regents, which includes several schools in addition to OSU. OU, several schools in addition to OSU. Like Cameron down in Lawton's covered under the OU system. Langston is covered under the OSU system.
Oof.
There's different things like that. And then Russo is all the regionals. And then there's a formula that was done in like the 80s, and it's a funding formula of how it gets divided out by everybody. There's been a big push to get that funding formula redone.
Yeah, it's 2026 now. That's up there.
Yes. So like that's where we want, but again, there's way more politics in education than you realize. And I didn't even know all of this until, this is years of me serving on faculty senate and being on the exec team, and years of me doing other things around the university to kind of piece together how this all goes. But the normal, definitely the student isn't going to know that this is how it all happens.
Well, and this is why I love asking these questions, and I'm so grateful that you're a wealth of knowledge in this, because I think it's important for us to know these things.
Yes, it's very important.
And for us, naturally curious people, it's just I don't know. I think it sort of lifts the veil a bit.
Absolutely.
Right? And
the numbers that we report, whether it's faculty to student ratios, all of these things are playing into funding and what's happening and how much research we're doing versus how much we're teaching and how that all goes. And I'm always a little bit surprised sometimes. I feel like we almost, for students, need a web page that just says, okay, here's how the hierarchy goes, because they don't you wouldn't necessarily understand, there's a difference in an assistant and an associate and a full professor versus a chair versus a dean versus a provost versus a president, right? And sometimes a student might be a little bit upset and they're like just straight up emails the provost, and you're going, well, that's great, now it's got to come down the chain like 3 different ways before I can fix your problem. But if you just have emailed me, I could have fixed your problem. That hasn't happened to me, luckily, but I've had others that it has happened to. I'm like, we would have fixed it if you would have just given us a chance.
Well, and so that's trying to,
That's so fascinating.
realize this vision of the research button, right? It's I don't want to step on anybody's toes. I don't want it to, and so, but spending quite a lot of time trying to figure this out. I've went and talked to so many people, and I just keep going around and nobody really seems to I don't know. I've just gone and talked to people who I thought would know, and they just didn't, and it's not their fault, right? But it's just
getting the right person.
Yeah, and
that's half the battle. It really is. It's, I hate to say it, it's not what you know, it's who you know sometimes. And that's why I always tell students, come ask me. I might not know the answer, but I can get you to somebody who will get you closer to knowing the answer.
Yeah.
So a lot of that is just, you're sending the right message to everybody to say, hey, go talk to your professors, go talk to people, because they're going to have an answer.
Yeah.
How it all works out.
Most definitely. Yeah. And, for someone who is thinking about going to academia, I think you should sort of start to think about the politics within it, because there is definitely some politics that's happening, right? And that's just sort of the way it kind of has to go in a way, right? You don't want things getting out of hand, right? You want there to be sort of a hierarchy. It helps create some cohesion, right, and some less chaos, right?
Well, and understanding the limitations of the system you're working within. So academics does not happen quickly. So if I want a curriculum change, let's just say I want to move my spectroscopy class from a 3,000 level to a 4,000 level, that is an 18-month process before I can do that. I have to go through my department. First of all, I have to make the change. Then I have to go through my department curriculum, then my college curriculum, then the university curriculum, and depending on how big the change is, then it has to go through Russo and sometimes OSHA, depending on what it is. Like a new program, that's all the way up through OSHA.
Yeah. Wow.
So those, you're moving at an academic pace at a lot of times. So you are looking at a year to implement changes at times.
Interesting. So I want to start sort of trying to wrap up this. We're almost at 3 hours now, which
I've enjoyed all of it.
Yes,
so thank you so much.
Me too.
I'm so fascinated by the entire topic.
Yes. So I guess one definitely thing I wanted to ask you is, what do you see a pivot at all for you in the future of your research, or do you like where it's going, or is there a field that you're very curious about, that you've always wanted to go down that rabbit hole?
So many times. When I was actually at NIH, I thought about, when I was trying to decide what I was going to do with the rest of my life, I was like, maybe I'll just go get a master's in biostats. Bioinformatics, actually, because Johns Hopkins was right down the road, and I was like, I could get an online master's. That would be a thing.
Yeah.
Because you're always like, well, that's kind of fascinating. Because I've always loved bioinformatics.
So the omics and stuff.
Yeah, the omics and stuff. Yeah, that's kind of where that came from. I didn't. I went in a different direction, but so. I go where my collaborators take me, which is what I love. I love that students can come in and go, hey, I'm interested in this. And as long as we're in the natural products realm somewhere, I can go with you. We have a wonderful colleague in recent hire in chemistry that does more computational stuff. And if I have a student come in and wants to go more modeling and things like that, I'm happy to work with him to go that way. I've always loved computational chemistry as well. It's a fun area to see how, what the computer says we can do versus what real life says we can do. So I'm up to whatever people want to do.
Yeah, and for me, Dr. Teo's research that he just published, right?
It's all in silico.
Yes, in silico. That's awesome.
And to me, the thought that you can have a good or a real contribution to society and to science with just this thing in front of it, that to me is just
back that up with a supercomputer, but then you'd be
but I can easily SSH into
exactly.
I'm working on P and OU supercomputer as we speak, so
exactly. I remember those days. Hit submit and walk away for 48 hours.
Yeah.
Pete and Schooner. What's the
Schooner.
Schooner?
Yeah, Schooner. Yep. But do you have any last questions or?
Actually, I lost it while you were talking about.
Okay.
Yeah, but it's okay. I can ask you later.
Yes, you always,
I think I had. Let's see here.
And you guys will put my email somewhere on this so people can contact me if
Most
they
definitely.
need to.
Yes.
Most
absolutely.
If there's anything else that you would want us to link to this, just send it my way.
Do you have any closing remarks?
I think we've covered a lot of things today, which is great. I love that.
Yeah.
No, I think it's great that you guys are doing this, and I love that you're encouraging students to get out there and just be involved. I mean, you only do college once, guys.
Yeah.
So, get out there, and whether it's research or whether it's, a student organization, like you said, find your people. You've got to. That's how you do it.
Yeah. Well, thank you so much for doing this. It's been a pleasure. It's been a privilege.
I have to say, this is my new favorite.
Yeah. We were just yesterday. We were like, how are we gonna top Dr. Teos? Because our talk with Dr. Oh man, it was
It's not a competition.
It was so
It's not a
good.
competition?
No,
It's not a competition?
and that's what we said to each other. It's
Yeah.
not a competition.
It's not a competition.
But man, this was really great.
Yeah.
Thank you so much.
Yeah.
No problem. Thank you
All
guys for
right.
having
Well,
me.
Until next time.
All right.
Oh, we don't need to do the clapping.
Okay.
Okay.
Awesome. Okay.